Um, all right, so Danny, how was your stream, by the way?
It was good. It was good. It was kind of framed around. There was a new PBS Eons video about B Rex. Do you know the story, Belent, with the Tyrannosaurus that some of my colleagues found from my old Museum of the Rockies? And it turns out that it seems this Tyrannosaurus was female,
Oh,
and she was getting ready to lay her eggs when she died. Um, the video was
no.
kind of about that and like sort of a follow up to that sort of thing. And so, but we talked all about like sex determination in different animals and like, um, X and Y chromosomes or Z and W chromosomes in birds, and then different kinds of critters like sheephead wrasse, these kinds of cool fishes that actually they're all born female, and then the largest ones grow up to be male later in life, and just all kinds of cool, weird, and interesting um, ways that like nature has found to, you know, uh, to get species to reproduce themselves. It's really neat stuff. So
It's I find it really
we're all over the place
confusing
today.
as well.
Yeah,
Like parthenogenesis, that is
yeah.
classic in insects of like how something like that might evolve. And I know in insects at least it turns out it's in part due to like bacteria called Wolbachia that make the
Really?
animal. Yeah, it's called Wolbachia, and it like for parasitic wasps, you'll have an all female population that'll keep reproducing and maintain
Uh
all
huh.
female. Like, occasionally they throw a male, but it's sterile, short lived, gone pretty rapidly.
What role does the bacterium have in that? That's
It it's so unknown that all you know is if you cure them of the Wolbachia,
uh huh.
then they start. It's like they weren't parthenogenic ever. They just start laying regular, and you start having males, and like no, nothing bad happened. It's
That's really interesting. Wow.
Yeah, and not enough is known. Like this Wolbachia bacteria causes also like brain abnormalities to form in like
Huh.
in flies, so they start puncturing holes in the brains. And so like in
Uh huh.
each insect, it seems to have like differing effects. Like that's a function of age, like how far
Uh
along
huh.
they are when they get infected, the infection load, and yeah, it could even make you parthenogenic, which you don't need males to reproduce.
Really interesting. I mean, there have been some high-profile cases of parthenogenesis in archosaurs recently. In California condors, there were some females that gave birth despite not having any males around in their enclosure, and it turns out the offspring are just like clones of their mothers. It's really interesting stuff.
Yeah, I
Um,
mean, yeah, I guess it's the same thing in insects. Like it's that clonal because there's no genetic addition, but
yeah,
you have to
that's
wonder,
awfully
right?
painful.
Then, like, is that like what other animals can do that as a function of environmental stress, and then what's the long term ramification of that, right? Like, what is
Right? Like, are the offspring actually viable, or are they going to be sterile, or like, what's the? Yeah, yeah,
or susceptibility too to infection?
yep, yep, yeah. Interesting stuff.
Yeah,
Yeah.
super cool, but it's very puzzling. Always very puzzling
How,
on that.
how has your stream been so far, Belinda? And give me a second. Let me close the door over here
Yeah, of
while
course,
you
of
while you
course.
talk about that.
Yeah, Hanny, welcome in. How's it going, Hanny? Can you evolve clones? I guess it Hanny, I think the parthenogenesis is more of like a last ditch effort to save the population rather than forward evolution, if that makes sense.
But yeah, how's your stream been so far, Belinda? What have you been getting up to here before I busted in?
It was we were doing some science art today. We're doing some Emperor's New Groove science art. So we're doing this scene here. And can y'all hear Danny? By the way, let me know, y'all, if you can. I can hear Danny just fine, but I'm getting some
I can
feet.
hear me too, but
Okay,
I'm here in the room with me, so yeah.
y'all can hear Danny. He's a bit quiet. All right, let me turn him up a little bit.
Okay. I'm an ant quiet according to Buttspot.
Dang it, butt
You know.
spot.
And by the way, I think I saw Ken here in the chat. Ken, how are you doing? Ken was one of my crewmates this summer in Wyoming and Utah, and also attended that conference that I kept talking about today in Salt Lake Research Conference back in June of last year. Good to see you, Ken. Yeah.
All right. Well, if there's a little bit of an echo, y'all, that's probably a guest star thing. Because I don't we sometimes have that as like it sounds a little bit echoey and then it randomly clears up later in the stream, and there's like I wouldn't have even pressed the button.
Oh, okay. If it might resolve itself, then maybe we shouldn't restart it. I'm checking right here. I've got the proper microphone and everything selected on my end.
Okay. Maybe let me send you another invite, maybe?
Yeah, okay. Let's do that. Should I go ahead and close this one?
Yeah, if you don't mind. We'll try again.
Yeah. I'll do that in 3, 2, 1.
Alright, let me go ahead, y'all. We'll refresh the cache as well. I don't think that's going to be the issue, but let's go ahead and give that a shot. Let's see here, just getting the right source.
Together, we can rule the galaxy.
Murph! Thank you for the resub. 1 second, Murph.
Just subscribed for 15 months.
Thank you for those 15 months, Murph. Hold on to your butts indeed. Let me refresh this cache. Alright, cache refreshed. Hopefully that'll fix things. And then let me invite Danny again. There we go. Murph, thank you for the resub. For 15 months, Murph. That's another piece of raw science.
Please remain seated and keep your arms and legs at all times.
Thank you, Murph. Tommy Ploticus. Alright. So let's see. Alright, any better, y'all? Now I can't hear you, Danny. I enjoy Murph is like worse. it's actually worse. Awesome. Thank y'all for the feedback. Apologies. It's the here we go. Maybe this time. All right.
Okay, I can hear you.
I can hear you.
Beautiful chat. Can you hear me?
Together, we can rule the galaxy.
And Matt, thank you for entering that 2-month resub.
Scientist just subscribed for 3 months. Thanks to Adgers for my sub gift.
Thank you very much, Matt, for that.
Echoing out. That's bizarre. It shouldn't be doing that.
And it's weird with the echo and the low volume too, because we've done in the past where we haven't had, like it will randomly kick in and then it'll like correct itself without us doing anything.
I think that's just a Twitch guest star thing.
Yeah.
I don't think there's much we can do about that at present. Yeah.
Let's see, I can lower my volume a little. It's weird because actually, Camellia, I have Danny actually maxed out today on in the guest star, and you're usually at half volume.
I haven't touched anything today either. This is the same exact setup I've always used.
Yeah.
Let me double check, make sure. Yeah, we're the same here in OBS. I'm using the OBS virtual camera.
Yep, same here.
Same.
Yeah.
Weird.
Yeah, and Echo's getting sloppy, and we haven't touched a thing, so it's usually.
It's a self-healing system. Gremlins.
Yeah, exactly. It's the tech gremlins.
Yeah.
But no, we've been we did some science art today. We're working on this scene from the Emperor's New Groove with our Fruit Fly and Aspergillus character, because people, nice. Tommy Ploticus in particular wanted one for Valentine's Day, so we're a little bit late, but of course we needed to have Tommy have a special Valentine's, right?
I like that. Yeah.
And so other than that, we did chat a little bit of science. I gave people a little bit of a preview of what's excited about. That we've got some really cool news stories coming up tonight.
Oh, yeah. I was talking a little bit about that as well. I'm excited to talk about your paper, Balint, if you're ready to dive into that, or
of course. Which one should we start with?
Let's start with the zebra finch, and I don't know. I'm wondering if this is as important as the press release and the abstract make it sound. Like, could this actually be a successor to CRISPR? Yeah, yeah.
Yeah.
Maybe give us a little bit of background on this, just as we start, and also the journal Nature Biotechnology. That's the same font as in the journal Nature. This is affiliated with the journal Nature, right?
Yeah, so this is that, it's like the Nature subsidiary, subgroup, publication group, like all under the same umbrella. And the reason this isn't in Nature proper, but versus the Nature Biotech, is because the study is much more along the lines of actually developing a new piece of technology, as the journal name suggests, versus like actively proving that something works. Like,
Gotcha.
I think if they would have gone in and done what they said that this thing can do, then maybe it would have been at a level of Nature, like in a living organism.
And maybe it will be. Maybe they've got a Nature paper cooking up right now.
Sometimes it's almost back to back. Like, you can co-submit the two papers and be like, "Hey, this is coming down the pipeline." They'll be like, "Well, this is incredible. Let's just do both." So to give y'all a little bit of a background, we've talked about CRISPR a lot, I think across both streams, as a means of genetic engineering, where you can go in if you have a matching sequence and break whatever you would like in a very like regimented way, where you know exactly where you're causing there to be a break in the DNA happening. But what's really, I think, interesting is if you want to insert a piece of DNA, it doesn't always work well. So you can still break the DNA, and there's these things called... Oh my goodness. Uwo's Lab. Welcome the heck in, y'all. How was your stream? Uwo, the new rebranded name. How was the stream? Welcome the heck in, y'all. Oh my goodness. Uwo's Lab. How was the stream, y'all? What were y'all up to today? Welcome on in. That's a massive Raiderino. Lurker Gene. We're talking about genetics. Welcome on in. Jake creates stuff. To the Max, the Stairfax. I love that name. Welcome in. Fanny Slam. That's a good name too. Soy Milk. Jake creates stuff. Forest Dweller. Welcome in. JC Rouser. Cake Magic. Goombus. Pink Punk Jelly. Oh my goodness. Albino Moose 308. Welcome on in. Dark Wabbits. Oh my god. I'm loving all these names, y'all. Welcome the heck in. Vassar. Uwo's Lab slightly more. I do like the rename. I was confused for a little bit.
Is this Uwo to Owo to Uwu?
Yes. Yes. There was a.
Makes a lot of sense.
There was
Makes a lot of sense.
A bunch of rats says actually it's Uwo Slab, as in his abs. So apparently the rename might be due to the abdominals. Ari the Fox, hello. Welcome on in, another first-time chatter. Uwo, what were y'all up to today? Tell us all the things. We want to know the details. Don't listen to them. I don't know. Now we're set on abs here. I'm just... Guys, Uwo's Lab does science, does Minecraft. A lot of really cool stuff. What were y'all up to today? And thank y'all for all the follows as well. We were trying to make the fastest burning candle.
Oh.
Did y'all succeed? I like that.
Sounds like it would have important industrial applications to, I mean, if we're trying to get people to buy more candles, then just get them to burn faster, you know?
It did burn
planned
really fast.
obsolescence right there.
And it worked. Nice. Like, Hardcore X Hunter says he made smoke. And then all of a sudden the comment from earlier, he made smoker's lungs, also now makes sense. I like that. Now you can speed run meditation. No. Uwo, no. That needs to be...
That's definitely the point, yeah.
Nice and gentle. But Raiders, welcome on in. You're catching us on a crossover day. So my name is Balent. I'm 1/3 of Science Streams. The other 2/3 are my wife Lita and our daughter baby Alona. This is Professor Danny Anduza of Paleontologizing. I got a
hey everybody, it's good to see you. Yeah.
Legendary, the single best science communicator on Twitch. His name is highlighted in the channel description. So y'all, if you are coming in, if you like science, please hit follow on Danny as well if you are enjoying the content. We cross over every Wednesday to talk about our respective fields. Danny is a real-life paleontologist. He talks every day on Twitch about the science that gets him excited and like publications and more. He even does field work on Twitch where you can see him dig up dinosaurs.
Live on stream. Yeah.
It's
tune in this summer for more of that. We're going to be in Wyoming and Utah, maybe Idaho as well this summer, digging up dinosaurs.
Nice. And actually, you did discover a new species of dinosaur last year on stream, right?
3 new species.
3 new species, yeah. So,
Yeah.
y'all, please tune in to Professor Anduza. My specialty, along with Lita's, we're both have our PhDs in molecular biology, and we do interactive science here on Twitch with microscopy. So we look at 30 million year old pieces of amber with insects and identify them on stream. We do model building. We do science art. We do science use of the week, deep dives to scientific topics. We do games, 3D printing, and we also do late Cretaceous fossil hunting we've been getting into lately, IRL streams. We've been looking for shark teeth and then identifying them under the microscope. Thank you all for the follow. So if you like science, please follow Danny. His name is highlighted in the chat below. And also give us a heart if y'all like the science. Again, we're chatting about our respective fields today. We always bring this new paper to each other, or new papers, and we chat about them from our unique perspective. So right now we're chatting about a new type of gene engineering that'll hopefully be an augmentation to CRISPR genetics. And what's really cool about this is derived from birds, which
Dinosaurs. Yes.
exactly, which actually was making. I know we can't get yet genetic information from dinosaurs, but given this finding, I was like, wow, this would like just the thought experiment of what this would mean for our knowledge of dinosaur genetics and jumping genes in dinosaurs. You know, and that's honestly, like, I saw the method, I was like, all right, the method's pretty cool, but I was like, oh no, this has birds in it. I think Danny's gonna like this, and so that's why I ended up picking it.
For anybody who might be new, just jumping in here, you're going to be a lot of very science-minded folks. You probably know this already, but for anybody who doesn't, there is a general consensus in the paleontological world, in the evolutionary biology world, in the ornithological world, among bird scientists, that birds are living dinosaurs. Birds are the only group of dinosaurs that survived the extinction event at the end of the Cretaceous period, when that asteroid hit the Earth. Birds are the only living descendants of dinosaurs. We know they evolved from small, two-legged, feathered, meat-eating dinosaurs, kind of like Velociraptor. And so, strictly speaking, birds never went completely extinct. We still have them today, and some of them are contributing to scientific research like this. It was the zebra finch, I think, that was used in this one, Bullet?
Yeah, and those are the, like, for a lab animal in terms of bird, it seems like those are the most common and the most well-known, so it's not that surprising that that's where they found this tool. And actually, we've got
a model organism,
Yeah, I think I got a good question from Area the Fox, and Danny, if you want to take this one, for gators and crocs.
Yeah, so alligators and crocodiles, crocodilians is that group name, they're kind of like cousins to the dinosaurs. So they split off before the first dinosaurs evolved. The way that we as scientists classify living things is based on their phylogeny, based on their evolutionary history, based on their family tree. And so, in order to be a dinosaur, you have to have evolved from the first dinosaur. And crocodiles and alligators didn't do that. They were on a similar branch, but they diverged before the first dinosaurs evolved. And so they're not dinosaurs themselves. But birds are, because birds evolved from dinosaurs that looked a lot like Velociraptor. Similar to, well, sharks are even further back. Sharks are, they're a kind of cartilaginous fish. Sharks and rays and chimeras and sawfish. They are, they're like way outside. They're barely even vertebrates. They are vertebrate animals, but they don't have bony skeletons like fishes and other tetrapods do, or like tetrapods and other fishes do. So yeah, in order to be a dinosaur, you have to have evolved from a dinosaur ancestor. And crocs and gators didn't do that. Not quite. They're very close, but think of them like cousins to the dinosaurs. And sharks are super far away. They're not anywhere close to dinosaurs.
And then, Danny, were dinos warm-blooded since birds are warm-blooded?
Ooh, that's an excellent question. This gets into something that we're going to talk about later, too, called phylogenetic bracketing. So, like, if you're trying to figure out something about dinosaurs that's hard to tell from just fossils, you look at their closest living relatives. So you look at birds, which are living dinosaurs, and then you look at their other closest living relatives, which are crocodiles and alligators. Like I said, they're like cousins to the dinosaurs. Birds today are warm-blooded. we've got direct evidence that a lot of dinosaurs are warm-blooded too. Seems like the closer you get to birds, the more warm-blooded you get. There are some dinosaurs that may have been closer to cold-blooded, but that might just have been because they were really big. And when you're a really big animal, you don't need to generate as much body heat. But for the most part, if you were to say that like most dinosaurs were warm-blooded, you'd be pretty close to the truth. That's the current consensus nowadays. If you're a dinosaur who is cold-blooded, you're kind of a weirdo. Like, that's probably a metabolism that you evolved like in a special weird way. Dinosaurs, the first dinosaurs were probably warm-blooded, we think. And then any ones that were cold-blooded later, that's something they specially evolved in a different direction to be cold-blooded, ectothermic. Yeah.
And that bracketing actually, I feel like, makes the argument in this study as well, because you can start to possibly infer what the ancestral state was. So for the raiders coming in, I don't know if y'all know about this CRISPR, which is a genetic technology that we have a command for because I always forget what it stands for, but it's clustered regularly interspaced short palindromic repeats. Essentially, it's a way to, and I know Nightbot like rude to me, telling me that words are hard, right? Shout out to the mods for putting that in. But it's a way to target a particular genetic element and to get rid of a mutation or to induce a stop. So if you want to interrupt a gene that you just want to turn off, you can go ahead and do that. And Hanny, CRISPR is discovered in bacteria, and it's part of their natural immune system. They actually insert genetic elements throughout their genome to then target other like bacterial or viral invaders, and that's just how they that's their immune system. It's just built in their genome, and they just essentially have this immune memory that's forever there that can be removed and then made into an mRNA, small RNA molecule that then is eaten up by the CRISPR, eaten, then matched elsewhere, and boom, you've taken out the threat. So it's actually we hijacked the immune system of a bacteria to do genetic engineering in any organism really that you have the imagination to use. Now, it's not that easy. Every time you have to optimize the protocol, but it is a really cool phenomenon to be able to do. Clam the Clown, thank you for the follow. Welcome in. And so this study's looking up at the follow-up to that, and that follow-up is genetic insertion. So the idea is, let's say you have a gene that is broken, where something is not working, right? So like, what do you do to make something work again? You have to insert something, and it turns out CRISPR is not especially great for it, or at least in a reliable way, where if you're trying to, because what you'd have to do is break the DNA open. They have to have these sticky ends, meaning that they're broken in a way that they can, pull up something. And then you have to make sure they don't just stick back to one another, but they actually pull in this new genetic element that you've put in. And then they have to, it has to be put in the right orientation, because apparently if you put it in the wrong way, it won't turn on. So there's a lot of extra factors at play. So the researchers here were looking for a way to insert a genetic element that was not already there before. And the goal being, let's say, if you have an organism that has something missing, you can put something back. And so it's not using CRISPR, it's using this other technology, and it's derived from birds. And so it's called a transposable element. And what that means,
a jumping gene almost, right?
Exactly. Yeah, exactly. It's a jumping gene. And so and apparently our genomes are full of residuals of this jumping gene, where if you were to call something a junk DNA, that's usually the closest thing it is, because it's residuals of these hopping elements. What they do is they copy themselves and then they insert elsewhere in the genome. And you can have two kinds. You can have ones where they just insert themselves multiple times, or you can have another one that removes itself and then inserts itself elsewhere in the genome. What that means is, sometimes they remove themselves and it's totally fine. They don't break anything. Other times, if they remove themselves, they actually break whatever gene they're next to or within. And by break, I mean that is now disrupted and you can't have a functional gene anymore. So they can actually trash the genomes if you're not careful. And what's really cool about this is there is this element called the R element, which recognizes this transposon and causes it to activate. It is in birds. The R element is not in mammals, but the receptor is.
Right, which kind of implies that mammals, at some point in our evolutionary history, we had this too, and we lost it somewhere along the line. Because mammals and reptiles, if you go back far enough, they do share a common ancestor at the base of the amniote clade. But we've been separate for a long, long time, like 350 million years or something like that. So we might be talking about that far back in time. The ancestors of mammals had this, and then we've since lost it.
And isn't it wild, to Danny, that we haven't lost the receptor?
Yeah.
Like, that's really,
it's
really
gotta
cool.
Mean that, like, I wonder if that, Blint, as a geneticist, does that imply to you that it does serve some other purpose, or it's just something that, like, it's part of the leftover architecture and it's not doing any harm, so it just sticks around? What's your first instinct there?
I mean, that's right. You would have thought that there would have been some selective loss for it, right? Because, or at least mutation in the receptor if there's nothing else binding to it. And
right.
Yeah, I would suggest to me that maybe somewhere else in the genome it's active. So where they looked at it in the nucleolus, which is, if you look in the nucleus of the cell, there's always this really, really dark circular region. And that's not the DNA, it's a bunch of ribosomal genomes hanging around in there. So it's like protein production that's what it's responsible for. And that's where this R element comes in and binds, and that's where it inserts whatever gene you want to put in.
Oh,
and so
interesting.
And what's cool about that, Danny, is like the ribosomal genes, there's tons of copies of them. And so if one of your genetic elements is put in, maybe you disrupt one of those ribosomal genes, but because there's so many copies, it doesn't have a result of the function, like, for the cell. So the cell is still able to have normally producing ribosomes. They still make protein and convert it, but you know exactly where you've put in this construct, versus a lot of jumping genes have no selectivity. They just kind of hop somewhere in the genome and you have no idea what you've messed up.
Sure, okay.
And Perilous 3 says it's important that this is all done in PCR. So this is not done in PCR. This is done in cell lines for this study. So this is this is all done in living cells. They are wanting to move on to another organism. They've done CRISPR genetic engineering, which is the precursor for this. I mean, I've done it in ants, but you can do it in, ants, flies, zebrafish. I think finches they've done too. They actually have done it in sickle cell patients. They cured it in sickle cell in people as a function of CRISPR. It's
really?
The treatment is like $10 million. You have to have the insurance of a prince, but it worked.
Yeah. Wow.
Yeah.
Wow.
Like it worked, as well as they've done some trials on vision and like correcting vision as a use of CRISPR. And so this is meant to augment those studies, and I think it's really, really exciting, because what this means is, and why it's easier. So with CRISPR, you need to induce a cut site in the genome, and then hope whatever you're trying to stick in actually gets put in. This does it all in one step. This inserts a genetic element in a known group of genes. So you're still disrupting a gene, which is why I think people are going to be a little bit hesitant about it, because you are potentially breaking something that is functional. But the justification they're making in the paper is it is only in these ribosomal genes. Like, those are the only area in the genome that has these receptors, and so it'll only be concentrated there. So as long as that's the scary part, you get the dosage right, you'll only disrupt that group of genes and like a low level of them, maybe one or two out of the, let's say, 50 copies that you have. And so it's a means to which putting back something that's broken. So let's say if you're missing, a gene for hemoglobin, like a correct copy of hemoglobin in sickle cell, instead of editing the cells the way they did in the previous treatment, each one, now it'll just be inserting a normal copy of the gene that'll get turned on and it'll work, and you'll be all set. What could go wrong? So Spartan, the dosage, right? It's still like jumping genes can be scary. Jaded, thank you for the lurk. The thing that helps, I think, is actually their strongest argument, and that's why I thought you'd find this interesting, Danny, is that it stems from birds. Is that if we had the receptors, if we have the functional transposable element, it would be in our bodies all the time. And birds have this all the time, right? And they're not trashing their genome. It is localized to only where the receptor is, which is only in one part of the nucleus, the nucleolus. And that is called a very resilient part of the nucleus, because there's so many copies of those genes that it almost gives you a buffer to build up mutation, and you end up being functional, like regardless if you build up a couple of mutations or not. If there were receptors everywhere in the genome, like most transposable elements, like there's this one called the line transposable element that can go anywhere in the genome, and that could trash your genome. Actually, Danny, this is weird, Danny. I would love to have your opinion on this. And Peerless, thank you for the follow. Fruit flies, up until the 50s, did not have transposable elements.
Together, we can rule
Peerless,
the
a
galaxy.
follow and a subscription. Oh my goodness, thank you for that.
Fantastic. Yeah.
P3 Realist 3
Welcome in.
subscribe.
A piece of raw science for you as well. For the subscription, thank you. Welcome in. And Purple Guy, thank you for the follow. So there were no...
Did I hear that correctly? Up until the 1950s, fruit flies did not have any transposons.
No. And like, something happened in the wild that it started spreading throughout the population. We don't know what it was. But if you look...
In the wild?
In the wild, yeah. So if you look at...
This is before people were modifying their genomes in a laboratory or anything like that, right?
Well, so in the lab, the first lab started around 1910 with fruit fly genetics, and they started doing mutagenesis studies where like you're just feeding them mutagens and like mutating certain things, right? So you got like the white-eyed mutant flies and wings, and even if you take those flies today, if you mate them with a wild fly who has transposable elements, that wild fly has repressors now in its genome to turn off those jumping genes, but your lab strain doesn't. And those jumping genes will trash its genome to the point where if a female hatches, it's sterile because its ovaries don't even develop. Because it is like...
Wow.
Yeah, so like how it evolved and how it got introduced into the population, still no one knows. But its evolution's amazing in that the wild ones have actually then now evolved a resilience against them.
This might be a little bit silly, but you say 1950s and mutagenesis, and my mind immediately goes to like American nuclear tests and Bikini Atoll and Godzilla and stuff like that. Because you know that we can, since the first atomic test in 1945, or 1944, excuse me, it was 44, right? When was that, chat? Was it 44 or 45?
I want to say
45,
40.
wasn't it?
Yeah.
Yeah. Like we can actually, we can look in the rock record. We can look in trees, and we can actually see the signature of those nuclear blasts in living things. Like if you cut down a tree that lived through the 1940s and 50s, you can actually see like radioactive isotopes in those that were introduced by those, by those nuclear tests. I'm assuming this probably doesn't have anything to do with our fruit flies. Like, it's probably not the same thing, but that's immediately where my mind goes to. Yeah,
and it's a great point, because it hasn't really been studied of what the connection is between those two. But it's just one of those like really interesting things that you can test nowadays in a lab. Like you can go in and mate them to wild flies and watch their genome just get trashed, versus the wild caught ones
really
have that
wild
resilience that evolved.
So in the wild populations, with nature red and tooth and claw and nature and all that, it's like there is selective pressures that caused certain populations to evolve those kind of like safeguards there that the captive bred populations, the laboratory strains, they don't have that.
Right, because there's no selective pressure. There's nothing in the lab that would make them need to be going towards that route, like, or selecting out the individuals that don't have that kind of protection. And then Spartan asks if we can grab the incidence of them geographically. Even island populations of species outside Drosophila melanogaster seem to have them.
Really?
Yeah, it's like
like you're talking about those ones in, was it Madagascar or?
Well, yeah, the Murciana in Madagascar, and then we had the Seychellia and the Seychelles Islands. Yeah.
Yeah, and they have those too.
Yeah, they have them as well.
Does this just sort of appear around the world at the same time in disparate populations?
Yeah. At least it's not as classified as I would want it to be, but it does seem like anything caught before that time doesn't have it, and species caught after that time do have it.
And it's not as though the technology to identify this didn't exist prior. It's that the thing just wasn't there prior.
Well, so even if you look at them now, those lab caught ones from the early 1900s, right?
Preserved specimens will not have that.
Well, so their lineage is still going.
Oh, I see what you mean.
Yeah, so the lab caught lines that we use originated one, and the one that I used a lot was from the 30s. And then that one, you can sequence it now, and it doesn't have any transposable elements throughout it, nor does it have those repressive elements that are in the wild populations.
Do we know absolutely for certain that wasn't a feature that was lost? Is there any possibility that those wild-caught ones originally had it, but then through selective breeding and just kind of having an easy life, these, flies chilling out in the laboratory, is there any chance that they lost that, and so we think that it's something that arose in the wild after, but...
That is a great point. There is some, preserved sample from, near the beginning, that when they sequence, it doesn't have those elements, but they also... They also caught multiple lines, right? And so I'm not convinced that there didn't just have one of the ones that are sequenced is a weird line in that respect. It could very easily have been maybe lost throughout time, but it's a lot of people who've done this work and they publish on it are convinced that they showed up, these transposable elements showed up later. And how it spread in the wild, there's not a good really satisfying answer of how it happened.
Wild.
Yeah.
That's really interesting. That's fascinating.
And also, my favorite part is that, how quickly it evolved resilience, and you can actually map the genetic elements that can, repress. Now, here are the repressors, and if you put them into the lab fly, and now you mate them, it's alright, everything's fine, and the genome is preserved, and it's not trashed, and... it's exciting, fun stuff, and a good population genetic look, and also evolution.
That's really cool.
And it ties back to this paper, chat, because in this paper, that could be what we were just describing in trashing the genome could happen if the transposable elements going towards a particular site of the genome, and here they are. It's all those transposable elements would be going directly towards that nucleolal structure. So it's ribosomal genome that's tons of copies of the ribosomal RNA and DNA, and disrupting one actually doesn't disrupt the overall protein level. And so it's it's a really smart and beautiful system, and I wish, Danny, they would have gone into how they discovered it. Because it just the paper just reads oh yeah, we tested this hypothesis and blah blah blah. There wasn't that eureka moment that went into it.
But there wasn't that moment, or they don't describe it in the paper?
They don't describe it. They don't describe it in the paper.
But you know that story and you say it's really cool, or you just wish...
No, I wish that they had. I wish that they had put something like that in there. But yeah, they had a nice figure set kind of demonstrating how classic CRISPR works. And I wanted to point out this to you, Danny, because I know you had a stream about this pretty recently, about AI used in figures.
Oh yeah.
And so this one uses BioRender, but they actually point out in the figure legend, and there's even a hot link to it of that's not an AI service, that is they have a pre-made figures and things you can use. You have to be the one curating it to move it around and make it look all nice and pretty.
And
but I thought it was
some actual human input and savvy in there, and not just some generative program or something.
Exactly. Yeah. Like it's
Yeah.
you know, like you had these lines in a DNA strand that were pre-built in a data set, and then you had to be the one to move them around, and it's different compared to that AI paper that you and I were both chatting about, which actually ended up getting retracted, that mouse one.
Why am I not surprised?
Well, you know, the sad part is if you read the journal's guidelines, they allow AI art.
Really?
Like, it's not against journal regulation to submit that, so it's frustrating because it'll reflect poorly on the authors.
Goodness. Did you see the, I don't know if I'll be able to find it here, but there was a thing going around on Twitter where somebody made, like, a cookie cutter that would make the, like, silhouette of that, like, AI picture of the mouse. It was really, really funny, but they, like, made cookies, I think, for their department or something.
Oh, no.
And it was blowing up on science Twitter. It was pretty good. I can't find it right now. But, yeah.
Yeah,
Oh, goodness.
And
Yeah.
Werewolf, I have the, I think it's this one here. Yeah, here we go. The original article. It was frustrating because it was retracted, even though, like, it turned out like no one actually read it. Like none of the authors
It's just a predatory
read.
journal, basically.
Yeah.
This is Frontiers, right?
Yeah,
Frontiers and nonsense.
well, they already have like really bad. They're the ones who sued that guy who was putting together like the list of journals that were for the predatory status. And so,
Oh yeah, you're talking about this.
yeah, Lita is like very actively angry, gung ho about them for that reason. Let's see, it's all right. Well, luckily I have the paper downloaded, so we still get to see it. Yeah, here you go, chat. This is the AI art that was made from it, and yeah, it was. The paper was had stat talking about mice, but it made a rat AI. So even the Spartan, the organism, was incorrect on this.
Yeah.
And in Perilous 3D, I feel the reason AI art is not beneficial on these is because AI is just taking from what we know, right? Like the system that we've already studied and we've identified. But if you're making new findings and new discoveries, if you're sweeping the internet as the AI, you're not going to be able to take into account all of these new findings. And it's just inaccessible, right? Like your goal here is to make science accessible, not just to people directly in your field, but people, field adjacent, like in science. And if you look at this figure, you're not going to get anything from this. Werewolf, that is the male reproductive organ of this rat.
Also, isn't a lot of the text on that just like garbled nonsense?
Oh yeah, no, it's misspelt. Even this one too, though, like this pathway is also the same way of just like.
Yeah.
It did turn out that the reviewers didn't actually read the paper, and then if you look, like, you would feel like the editor would be a little bit like it's from a little bit of like a respected institution, but that doesn't matter sometimes either. Like it's just people paying to get published, and then you get this out, and it just my biggest thing was was frustrating to me was in the journal itself, the guidelines said AI art is okay. Right, and so it's like, whose fault? I mean, sure, shame on the author still for submitting this. But
that's a bad sign when your journal is like, they explicitly state that that's okay. Especially when scientific illustrators are typically, they find such a difficult time actually getting paid for their work and making a living when that sort of work is really, really important. And now there's further forces arrayed against them. Like, I know I've got friends and colleagues in the paleo art community, and it's bad, and it's getting worse because of stuff like this, And
it's those artists are really good at what they do, and like conveying the information you're trying to demonstrate, especially in like a review paper, right, where you're trying to synthesize all the known work. Like, you can't have something like difficult to read like this. Like, you have to be good at it, what you do. And I just kind of this was like a hat's tip in this paper for me, is that, you know, they didn't make these, but they use BioRender, which is still requires human curation. They mentioned that in the figure legend. It just kind of made me think about.
Is BioRender like a? Is it basically like a? This is going to sound disparaging, even though it's not supposed to be. Is it like a clip art warehouse sort of a deal, where it's like there are good scientific illustrations that you can like take from this source? Is that kind of what it's like?
Yeah, it's like, think of it like a mix between clip art and PowerPoint.
Okay.
So like,
yeah,
you can search. There's a search feature, and you're like, okay, I want Drosophila, and there's like a pre-made Drosophila image, and then you can like put that, and so you can build your entire figure in their software, and then if you pay a fee, it'll remove the watermark, and you can go ahead and get it. Oh, I think Werewolf found the cookie cutter. There you go.
Oh, that's it right there. Holy cow, Werewolf. That's the rat cookie, yes.
Oh my god, that is brilliant.
Isn't that great? That's wonderful, yeah.
It's the homage, right? Now we have to... God.
Yep.
It's also frustrating, though, that like this got so much news as well. Like, it was trending so much versus like really beautiful figures, what people put so much time and effort into it and could really convey the science really well, just like isn't.
Well, I mean, to be fair, it did get a lot of attention because people were so outraged over it, you know, rightfully so. And hopefully this will turn people off of the idea of like, just use AI to make the figures. Nobody cares. Like, no, people definitely care, and it's not something to emulate.
Yeah.
But, and it's funny that what was the? Oh, sorry. Go ahead.
Oh, Nelf was just wondering the difference, like if they take out the Illustrator in BioRender, but Nelf, like the base figures are still made by a scientific illustrator. Actually, they hire sometimes like actual scientists to build the models that you can then use in the software.
We have a similar thing in the biological or in the like zoological sciences called Philopic. I just put a link into the chat right there. But when we're using, like when we're making cladograms, like an evolutionary family tree, this is where you can get scientifically rigorous silhouettes of different organisms in order to place in there. And I've actually thought of producing some of these live on stream, like making some silhouettes of dinosaurs that are not represented on there yet.
That would be really cool.
That might be something I do in the future. But yeah, it's just a resource for scientists to be able to use, and that, like, you can have a graphical representation of a certain critter, and sometimes there'll be 50 of these in the same figure if you're making a big tree. But it's just really nice to be able to have that as a resource and actually have it be legit, you know?
I mean, to me, the visual figures make a world of a difference if you're trying to articulate any one of your points, because otherwise, what are you actually trying to tell, right? If you're not making it accessible.
And imagine if you're like somebody for whom English is not your first language, and you're trying to make sense of a figure, and like this, it can really help out with that kind of thing, I suppose. Yeah.
And sleep well, Harissa. Thank you for hanging with us. You can't stay up any longer. Get some rest, Harissa. I appreciate you. So I didn't mean to rabbit hole too far into it, but I just remembered you were talking about on your stream, and we were talking about it here as well, about the AI art, and I was like, this is kind of like a nice in-between where they're using BioRender. It's not hand-making it, but it is making it from the like a scientist, and they have to piece things together. It's still legit.
It still has, like, actual human input and expertise that goes into it, instead of just, like, having some generative model fart out something that, hallucinate something, basically.
Exactly. Yeah. Fishman, sleep well as well. Thank you for the raid earlier tonight. And hello, welcome in, Cliff Alistair McLean. Cliff, how's it going? Welcome. Josephine Neal 128 and Jinx Coffee also, thank you all for the follows. Welcome on in. I thought the other thing I really liked actually about this paper, Danny, in terms of like data reporting is that they ran a bunch of gels to make sure that when they were inserting a genetic element back, that it was functional. So the idea was, okay, is the genetic element that we're putting into a genome using this jumping gene technology from birds, is it going in? So like if we sequence it, is it there, yes or no? And it is, which is great, but if it's non-functional, then no one's going to care. And so they ran these protein gels where the idea is, okay, if you've put it back in, there should be a band that is now present where it was once absent.
Right.
And what I was really hats off to me was in the supplement, you see here there's cuts. And there's like this 100 length, and then everything goes across here. I'll zoom in a little more. Basically, this first signal here is your ladder. And that gives you the size element to everything. And then afterwards, right, you cut and make sure that, you're just showing off selectively. And what I really liked about what they did in this study is in the supplement, they were like, here is the entire gel. Uncut. You can see all the things on there, and we're not just showing you what we want you to see, but we're showing you pretty much everything that we think you should see.
Right.
And I thought that was really impressive that they did that.
It's cool stuff. It's really cool stuff. And it's neat to hear that from your perspective, too, because I don't recognize how cool that is. I'm not part of this field. So to hear you explain that is really helpful.
I guess it's like one of those things where, you tell me what I'm supposed to be looking for. Like, they're like, you have bands that are important at 21, 60, and 100. And I'm telling you to look here. And I might be like, well, what about elsewhere on the gel? Like, are there other signals that are popping up and that maybe you're misguiding me? And so you can actually now look at the supplement, like, here's the whole gel. Right. And you can look back within the first figure and then this figure and see it's even telling you matching. Like, this is the entire gel for 1D. And you can see, like, it's not... Because what used to happen in the past is people would crop their gels. And it'd be really messy. And the rest of it had a bunch of schmutz. But if you crop it right where you want, it's like, well, it can almost be misleading. But by having the entire gel, it's like, no, you can look at it rest and your conclusion will still be the same. It just makes for a prettier figure in the paper.
Yeah. That makes sense.
And then, they had a bunch of gels kind of proving their points, and how, here is the outline for, we had asked from Perilous 3D. This is what they used for, like, they used cell lines to test this. And what I thought was really cool was there was this study here where they did cell sorting. So when all they wanted to do at first was, can we make these cells glow? So the genetic element that they were inserting was glow. Right? And it's like a very simple thing. But they had this cell sorter. And the idea is if it's working, you should start to see a shift in the population of cells. So for example, here in this first column, you see there's very, very few cells in this box. And then as you're increasing concentrations in either direction, you start to see populations forming in the top right corner of this box. And those are your new converted cells, like they're expressing this glowing protein. And so it's just showing, like, with increasing concentrations of whatever elements that you're putting in, you can change these populations and really shift them. And, like, that's like a really easy part. And yeah, Dame Karen, it does look cool. But it's like, it's just a fundamental, right? Like here, it's an easy test. It's a machine you just put them in. If it worked, like, honestly, like, it's the setup of getting the elements into the cells, but then you hook it up to the machine, and that's like, like 2 hours. And then you get these kind of graphs out. And so, this is kind of the first step that they were showing, like, yes, we put in glowing elements, it can glow, perfect. And then they went to, like, protein expression that's not just glowing, but something actually much more complicated and functional. And they had a nice summary figure kind of outlining, see, I may have just passed it, like what the methodology is. So it's essentially what they're doing is their DNA of choice will bind to a given site. There is this cut that forms from the transposable element, and then your DNA of choice inserts itself into the genome. And that's like the nice way of doing it. And again, it's nothing revolutionary. What's revolutionary is that it's only targeting areas with a particular receptor. Birds have this receptor, we have this receptor, but we don't have the genetic element in our genome. And so we're putting that in now in order to deliver some kind of mechanism, and hopefully as a cure for something. And or I guess it could be a genetic tool as well if you're trying to test something. So I thought this was cool from two perspectives. One is the possibility of, treatments, but also from the evolutionary perspective. Like, that means that our common ancestor, right, had this element, because we both as mammals have the receptor, same as birds. Where did that jumping element go? And what do you think, Dan, this is what I wanted your thoughts on, mean for dinosaurs? Like, what do we think that their state was look like? And I tried looking up to know if we have anything about crocodilians as kind of like that bracketing that we were talking about. And I couldn't find any info on that, unfortunately.
Yeah, I don't think there's been a lot of genetic testing of crocodilians in general. Because they're harder to keep in a laboratory. Like, alligators are tougher to keep in a lab setting than zebra finches. This does imply pretty strongly that dinosaurs would have had a very similar kind of thing going on as well. But where along that line did it evolve? Like, if we could look at crocodilians, say look at alligators and see if they have it, then that would imply that it evolved even before the first dinosaurs, and kind of the common ancestor of crocodilians and dinosaurs. Because if we have it in birds, which are like the end state for dinosaurs, and crocodilians, that implies that it goes back to their common ancestor. But yeah, if you weren't able to find anything about crocodilians there, then I guess we don't know. But that's the way to test it for sure. Yeah.
I guess it goes back to your point of what else might those receptors be used for? Right, like is it a later thing that evolved in our modern day birds, and the ancestral state used it for something else, or did it evolve, in like certain dinosaurs, and then like it's maintained in birds, and how that works? And until we can bracket it, we just don't know.
Right, exactly. Yeah.
And we actually, what's weird is we don't know the function of this either. Like, is there a function like there is with immunity for bacteria, or is this one theory from the paper suggests it might just be a jumping gene that's specific to birds? But it seems like, given that there's only receptors to it in one part of their genome, it almost seems like there might be some kind of functional element to it as well. It's not just like an invasive genetic element that's randomly coming in.
Yeah. I don't know. That's a really good question, and like, it's also a little slightly frustrating to think about, because whoever's doing these, like, nobody's gonna, I think, run a test to see if it's also in crocodilians to try and answer this question. We're talking about it because we like dinosaurs and we're fascinated by that, but like, that's so tangential to what this study was about, and nobody's going to investigate that, I don't think.
Yeah, I was kind of disappointed there.
It's not going to be their goal.
Well, there wasn't, like, I feel like it's ripe for talking about evolution, but it was very, it was like barely mentioned, like, here's the receptors and that's why it might have something to do with evolution. But then, I mean, part of it's a Nature paper, you're limited on tech space, and they're trying to push the tech element to it. Which, again, you asked me at the beginning if this is going to replace or like rival CRISPR. It's really hard to say. I think it has a lot of potential, but it's gonna be like, what does the living organism say? Like, they're gonna have to, I think, repeat the same glowing protein experiment in an insect, let's say, and see how well it works. Like, because the issue too with CRISPR and helping humans is, how can you hit every cell that you need to hit? Right.
Like, you have to do it like kind of one by one with CRISPR, right? And the idea with this is that you can kind of do it almost not quite genome wide, but how is this different?
So this is still one by one as well. It's just if you're inserting something with CRISPR, when you insert something, you have to break the DNA. And so you have a couple of things that can happen. One is, if you break it, you have your genetic element you want to be put in. And that's great, because that's your goal.
Yeah.
Or it fuses back, and there's no fix, or like nothing's gone in, or it goes back in but it's in the wrong orientation, and so it's non-functional. And so there's like all these possible things that could go wrong. And so even if you do miraculously hit every cell with your genetic element, you're still going to be a mosaic. This one, they're arguing you still have to hit every cell. You're still probably going to be a mosaic, but when it does hit, the likelihood of inserting that genetic element that could fix something is higher.
Gotcha. So there's less room for mistakes, for it to be inserted incorrectly or flipped or whatever.
Yeah, and
Is that
so
kind of the idea?
it's more... CRISPR's great for that initial like disruption of a gene, and then this would be for an insertion of a gene in a known area. And that's, I think, the big catch. Like, other retrotransposons had sites, but they're throughout the genome, and you could always make the argument, like, if that thing inserts in the wrong location and turns off a critical gene, like, that's death for the person, right? Versus here, if it inserts in one of those ribosomal genes, there's so many extra copies that it won't matter. Is the goal and how they're trying to present.
Gotcha.
It's still a little scary to me because, like, you know the region of the genome it's going to insert, but you can't tell me where it's going to insert, right? Like, you know it's in the ribosomal genetic cluster because of where the receptors are, but you still don't know where it's going in. And so I think that might make people a little hesitant. But they did say in the end of the paper and in the interview, like, we need to do further testing in an animal to see, like, what the next steps are. But I think there's reason to be optimistic for it.
Gotcha. Yeah, okay. Cool. Very cool. No, I was wondering about this too. The paper that I picked for today is kind of a methods paper, or like would you call this a methods paper too, from your perspective?
Yeah. Because it's for the methods papers that I've seen in the past. It's like we're doing something new, but we're like in terms of the method, but the question we're answering is something that's already been answered a lot of times. So you have expected outputs, and so like here, putting GFP in a cell, that green fluorescent protein. You know what's going to happen to the cell. It shouldn't result in cell death. You know all the outputs that could occur. And so if everything occurs, you do your method and someone else's, and your new method works just as well and doesn't have any side effects, and you're kind of like, all right, this new method is doing, what it's supposed to.
Neat. And I wanted to ask you, too, kind of getting into the more like meta science of this. Do you know any of the authors on this paper, or like, are you familiar with their work from the past, or their reputation, or anything like that? I feel like that's fun to discuss, and like while we're here talking with a geneticist, we should probably get into that.
Yeah. For some reason, Kathleen Collins' name is ringing a bell, but I can't.
I feel like I've heard her name before too.
Yeah.
And I think she works in Berkeley.
Yep, she does.
In fact. Yeah. So the building that I'm in, usually a couple times a week, the Museum of Paleontology at Berkeley, it's like the same building. Or I think the genetics building might be an annex to the life sciences building, but it's like, this is my neck of the woods. This is, yeah.
Let's take a look and see, like, what else she's done. Because, see, she's a biophysicist. Oh, telomeres, that's why. Primarily telomeres, so probably some aging effects that she's been looking at. Molecular and cellular biology, telomere maintenance, assembly of RNA. Yeah, they do this NICT prime DNA synthesis, which is like that cut site going in, which is essentially what that retrotransposon is doing.
Yeah.
Oh yeah, here we go. Like, here's her stuff in the news too. Oh,
nice.
She's in the Academy of Arts and Sciences. Aging and cancer. Yeah, that's probably why her name kind of hit. I was like, oh yeah, this feels like she's a big shot. Like, for some reason, like, and yeah, it's a lot of in the age. I know we keep coming back to aging papers, Danny. I'm sorry, it's like a theme that I keep doing.
Yeah, it's good stuff, and it's something we're all concerned about.
Yeah. But I thought it was cool with the birds. And Hanny asked, like, what diseases could be cured by ribosomal genes. So Hanny, in this study, they're not doing anything with the ribosomal genes in terms of like that isn't like the goal. They're inserting genes in that region of the genome where it's just copies upon copies of ribosomal genes, the same genes. So if you insert a genetic element there and it disrupts the ribosomal gene, the idea is it won't affect the cell because the other ribosomal genes are still functional. And so you mess up one, but you still have 9 that are functional, and so you're okay. So it's, imagine if you had sickle cell, so non-functioning hemoglobin, you put in a functioning hemoglobin gene into that region of the ribosomal genes. Yeah, you turn off one ribosomal gene of where it lands, but everything else is still functional. So you give the human functional hemoglobin, and then the ribosomal genes are in theory still activated. The catch is, as Danny says, are we going to hit every cell? Not even, I mean, with this tech either, that's not going to happen. And
could you do that very early on in an embryo or something like that, though?
So that gets into ethics, though, right?
Yeah.
Like, how
yeah,
does,
for sure. Yeah.
like, because if that's actually how we do CRISPR in an insect and in a mouse too. Like, you want to hit as early in development as you can, because hitting 1 cell or 3 cells is much easier than, you know, a billion cells.
Right.
And there's that study.
This would be heritable through different cells. So, like, if you can do it very, very early on where you've just got a tiny cluster of cells, these differences, like, those modifications will be passed along through that cell line until you've got the full organism. Is that the idea?
Yeah, and the biggest thing, like,
Okay.
in the research side of things, like, not when trying to cure a patient, but making a lineage that you can study, is you want to make sure that the reproductive system of that individual also brings up this genetic information so you can breed a line.
Right.
Obviously, for a human, we're not trying to breed anything, but let's say if we needed to someday give genetic resilience to an embryo because we're, like, our population's getting wiped out because of some virus, right? You would even want that human to be able to pass on that resistance gene, and then you'd have to early on in the embryo. This is like really crazy sci-fi stuff, but that would be like a similar idea behind it.
It gets into, not to sidetrack us too much here, but there's that company Colossal Biosciences, and they're talking about kind of resurrecting the dodo through cloning. But the thing is, we've not been able to clone birds so far for a number of different reasons. Like, nobody's been able to ever clone a bird. Their eggs are too big is one part of it, and the embryo starts off too small compared to the size of the egg, as I understand it. But the idea was that they would take a bird like the closest relative they could get to a dodo, like I think it's a solitaire pigeon or something like that, Rodriguez solitaire, and they would try and modify it so that it basically produces dodo eggs. So, like, just modifying the reproductive system of this Rodriguez solitaire so that they can introduce certain genes. So it's basically just producing, like, they're able to take if they could fully sequence the dodo genome and then insert that into a cell, they could get this thing to just produce dodo eggs. That's kind of the idea there.
So, like,
is that kind of what you're talking about, where, like, you're modifying the the parts that are reproducing the new offspring?
So for for an insect, I guess it works a little bit differently because they're stem cells, and those keep dividing, so you don't have a set number of eggs. You have unlimited eggs as long as the stem cells keep dividing. So those are the reproductive cells of the animal. And so you want those original cells to have taken up this new genetic information that you've inserted, so that every time they divide, it will have that new element. So every member of that offspring will have that element to it.
That makes sense. Yeah.
And so that's like a problem with ants, because you can't mate them in the lab. And so every time you want to do like a genetic perturbation, you have to do it every time on a worker, right?
Yeah.
Hope the worker doesn't die. And then you have to measure how much you've changed the genetic element in them, because it's not standard, because every time you're doing a new injection, and so it's that complicating factor. But like with mice, you know, you hit the 4-cell stage in a mouse, you mutate those 4 cells. At 6 cells you take out, or 8 cells you take out one of those cells to sequence, see if that cell has taken up your genetic element. If it has, the idea is that the others will have also. You let the mouse come to term, and then you can, you do like a little snip and get a blood sample from the pup and see. You sequence that and be like, okay, are all the cells here acquired this genetic mutation? And you usually have. If the 7-8 cell stage has it, then usually the adult has it too.
Huh.
And then you just breed them, and they keep passing on that genetic information.
Right. Whereas, like, for a bird, it gets more complicated because you've got to alter the ovaries of the bird in that.
Yeah, it's like...
Is that making any sense?
No, well, yeah, it's like with mammals, you have a set number of eggs, right? So after the animal divides, you're done. Like, everything gets divided, you're set. Versus the birds, I feel like... I don't know enough about bird oogenesis, but I think
they have a set number of egg cells like mammals. I kind of doubt it, but I'm not totally sure.
For some reason, I have a neuron find that they don't. In which case, you'd have to hit, again, the right cell population that's leading to the bird eggs being produced. I think it's one of those that should be possible, but one of the hardest part of CRISPR genetic engineering is knowing when to do those injections and how to test that you've gotten it. Because with the more complicated you move up, it takes longer to know if the experiment worked, right? Like, coming to term for a bird is longer than an insect, and then sexual maturity, and then mating them, and making sure it's heritable, and then establishing a male and female line where you can keep mating them, that it's heritable, and it's not causing problems. Like, it's
like orders of magnitude more complicated.
Yeah. And it's one of those where, like, in labs, people will be like, well, why don't you just CRISPR it? Like, it's just, you just use it as a verb. It's like, well, because it could be really, really difficult to do that and quite terrible. Yeah.
That makes sense. Yeah.
And Perilous says, you would have thought after the Dolly experiment, but as soon as I thought about it, realized it only worked as a clone of existing. If you take the existing clone, like, that's something you can pass on. Actually, like, they've de-extincted, if that's a verb now, like, the black-footed ferret using genes from... Yeah, with the Elizabeth Ann. Oh, we got a raid in from Astronomy Show. Astronomy Show, welcome the heck in. How was your stream? Y'all, if you don't know Astronomy Show, another science and tech streamer here on Twitch who does live astrophotography, as the name would suggest. Go check out our friend Astronomy Show, aka Jason. How was the stream, my friend? I hope it was amazing. I hope you all saw some beautiful sights. He actually, Danny, Astronomy Show had a clip that went really viral because he caught, like, a shooting star, like, on cam, and it blew up to, like, there was, like, thousands of people watching this clip, which was really, really cool to see, you know, one of our science friends, like, doing the astrophotography and, you know, really taking off. Astronomy Show, I hope you had a great stream. Welcome the heck in, everybody. My name is Balint. I'm 1/3 of Science Streams. Other 2/3 are my lovely wife, Lena, and our daughter, baby Alona. Research scientists. We do live streams here on Twitch 7 days a week with science. We have a special guest on our Wednesdays, Professor Danny Anduza, single greatest science communicator on Twitch and YouTube's and the internet and in person. Paleontologist extraordinaire. Please go drop him a follow. His name is in the channel description, and we'll also be giving him the fancy pants shout out when the cooldown is done. Television warrior, welcome in. He was educating me, a simple man, about the wonders of the moon. Nice, very cool.
Nice. Moon's pretty cool. Yeah.
And yeah, Astronomy Show. If you want to drop in the link to the video that went viral, so Werewolf Cast and others can see, feel free to drop it on in. We were just talking about a new genetic method for inserting genetic elements from bird genomes. It was taken to be used in mammals to hopefully lead to human cures, but also manipulate the genome. And I think we were just doing our wrap up on that thought, and then Danny, if you're ready to cross over to your methods paper, which is funny that we both picked a methods paper this week as our primary.
I know, right?
one.
It's all about the methods this week. Before we do that shoot, I wanted to ask, Lynn, do you think that, you know, they're always talking about human applications for stuff like this? Would this particular method described in your paper that you selected, would this be useful for altering the genomes of birds? Can we kind of skip a step there? And,
like
or
a means of genetic engineering in the birds. FF Matters, thank you for the follow. Welcome in. I think why not? Because even in this study, they suggested that the receptor in the birds is also in that nucleolar region. So just the nucleolus. And to me, that would mean that you can still insert things into the genome.
Okay.
And so you could maybe insert, you'd have to test it the same way they did in this paper on cells, like put in green fluorescent protein, see if you can get it to glow. You do, the catch is like you need to have what are called drivers. So that means when you insert GFP, that green fluorescent protein, you have to add in something that tells where that protein should be turned on. So like the factor that tells you this should be on in neurons, or the factor that says it should be on in heart cells. Because otherwise, right, it's in every cell. How do you know where to turn it on? And so I don't know if we know enough yet about bird genomes to be able to have that additional piece on. I mean, we would, if you want it everywhere, you can put an actin thing on there because you have actin cytoskeletal, filament in every single cell. So you can turn it on in every cell with that because every cell has actin. But if you just wanted it, let's say, in the ovary, like for bringing back the dodo, I don't know if we know well enough that what just turns on in a dodo's over, or like in an ovary of a bird, and it might be different between different bird species. But I think it could. I think it could. I wonder if Jack Horner's chicken project could use this. As like because I know they're trying to, like revert. As I love it, he says in the
engineer a velociraptor like. Maniraptoran from a chicken,
Yeah, I really like it in the that TED talk where he, about the kids and like showing the kids about like no one cares about the chicken.
He's like my whole point about this is like I'd love to be able to walk on stage with a dino chicken and be able to say, what is this?
Yeah.
Is it a bird? Is it a dinosaur? It's both. Just like being able to show people rather than just telling them would be really, really cool. And and get a lot of people really fired up about science, about evolution.
Absolutely. And Ask a Scientist Gaming, thank you for the follow. Yeah, Danny, let me show you. This is the clip from astronomy show in the night sky. See right there? Right in the center. Oh, we'll replay it.
Oh, beautiful. Nice.
Yeah.
That's really cool. That's really cool astronomy show. That's awesome. I can tell you the first time I ever saw a shooting star, I was in high school because I grew up in the San Francisco Bay Area, pretty urban environment, lots of light pollution. I'd never seen a shooting star before. The first time I ever saw one was at like an outdoor ed camp, and it was like really, really dark out, and I'm just looking up at the stars, and then just whoosh, and it really scared me at the time. Just like this deep, visceral dread. And I think the reason I had that reaction is A, I'd never seen them before, but B, growing up being really interested in dinosaurs, I associate, big things streaking across the sky like that with like mass extinction. And so just instinctually, or I guess not instinctually, it's a learned fear, but like, it really scared me.
And because you've said too, like you wanted to be a dinosaur as a kid, right? And then you look up and you see, oh, good God, that would be terrifying.
Yeah. Oh man. But yeah. Anyway, speaking of dinosaurs, should we get into that other paper?
Yes. So, this is such a cool paper.
I'm glad you liked it. Yeah, we talked about this briefly on my stream the other day.
And I have the videos too, Danny. I downloaded the supplement.
Let's start with that.
Okay.
Yeah, great idea.
Do we want, because there's this one from Yale that they put out. Do we want to watch that, or do we want like from the paper itself?
Let's take a look at this one first,
okay,
just to kind of frame the conversation. I think chat would appreciate that.
Okay, let me put on the captions. Here we go.
For centuries now, if paleontologists wanted to figure out how extinct animals moved, they had to pick up fossil bones, move them with their hands, and try to get a sense of how they would have fit together at the joints of the animal. But they were just kind of going on intuition. They had to say whether things looked right or looked wrong, and they had no data to back up that intuition. So what we've done here...
I feel like, Danny, in talking to you, like in all these
Uh-huh.
crossovers and in your streams, I feel like that part isn't exactly accurate.
Yeah, it's selling us short to say that it's mostly based on intuition. Like, sometimes, I don't know, do you ever have this, Belinda, in your field where people try and quantify things where you don't necessarily need quantification? Where,
More and more.
yeah, it's almost like a physics envy kind of a thing, or something like that, where it's like you want to have numbers on a certain finding when it's like, well, it's more of a qualitative kind of thing.
Yeah, it's become one where it's very much going against the grind to have anything just qualitative. Like, even if
Yeah.
it's something where it's like you look at it and it's clearly black or white,
Uh-huh.
it's much more of like this, like, you almost have to invent a way to quantify it now, which is more convoluted than just saying, like, here are the pictures, like, you go ahead and take a look at it, and it's, like, much easier to see it that way.
Yeah, I don't think that this paper is there, but I'm my heckles are already up a little bit. Like, you know, I'm like I hope that this is not going in that direction, because we do have stuff like that in paleontology where it's like, oh yeah, I got to put a number on everything, when it's very much a garbage in, garbage out thing, and there's a lot of uncertainty associated with those numbers in the first place, so it gives us like a sense of false objectivity, maybe.
Why do you think
I
that
don't
is?
think that's what this paper is doing.
Like,
Yeah.
why do you think that's become a, I guess maybe even a more common thing? Like, or
I
like,
think,
like
well,
that
a lot of it is
mindset.
in cladistics. So, like, cladistics was revolutionary for paleontology back in the 80s. For those of you who are in chat wondering what cladistics is, it's like trying to make family trees of different organisms by finding characteristics of those organisms and then coding them as different numbers. And then you run those numbers through a computer, and then it should tell you who is more closely related to whom, and it'll basically spit out a family tree for you. Call that a cladogram. But the thing is, those numbers that you assign to them can be really arbitrary in the first place. And there are ways to, like, kind of test these methods to make sure that you're getting something real out of it, but a lot of people don't bother with that, and sometimes it's really tricky. And I don't know, anytime a computer spits out a result, I think there's kind of a natural human tendency to go, oh, well, the computer says it, so it must be right. Where it's like, no, you told the computer to say that. You may not realize that you told the computer to say that, but you did. And it's a big problem in vertebrate paleontology, especially, I think. Yeah, because I think a lot of paleontologists, they don't come from a computer science background. I certainly don't, and that's why I'm a little bit suspicious of stuff like this. But, like, sometimes people go in the opposite direction, where they're like, "Oh, well, the computer said it, must be right. The computer says so." Does that make sense?
Yeah, and I like how you say, you know, it's that garbage in, garbage out mentality. Your data is only going to be as good as
Yeah.
what you point in. So, like, for us, it's like the
Yep.
model trainings of gene predictions, and you're trying to predict
Uh-huh.
new genes that act in a similar way. That's you might get a list of genes, but they could all be garbage if you didn't build the model correctly to begin with.
Absolutely.
Yeah.
And so anytime that I see like tried and true methods being replaced or usurped by more quantitative methods, I'm a little bit suspicious. But it seems like they're doing stuff right in this paper. A lot of this flew straight over my head. But I'm glad you liked it, and I'm excited to talk about it.
Yeah,
So,
I'm getting excited, just like jumping. Let's continue on in the video and set the frame for people.
Yeah.
Is we've taken x-ray videos of birds walking. We've looked inside their joints to see how the shapes of their bones fit together, and we've used the information we've gotten there to come up with a way to calculate a mathematical score for how joint surfaces fit together. We then took what we learned from the living dinosaurs and we applied them to the extinct dinosaur Deinonychus.
I like how they said the living dinosaurs, by the way. It was like a little
yep.
slid in there, and also the fact that it was Deinonychus. I thought that was an interesting dino choice to do.
Well, it's kind of one of the few dinosaur choices they could do at Yale, because they don't have a lot of other stuff.
Oh, so I thought it was like some kind of like public perception thing that they wanted to do, and
It's
not
very much a Yale
accessibility.
thing, no, because they've got the holotype of Deinonychus there at the Yale Peabody Museum.
Gotcha.
And that's really the only. It's basically like, I don't know. Yale started off during the Bone Wars with Othniel Charles Marsh. So that his uncle, wealthy uncle George Peabody, the great American financier and robber baron, he basically gave his nephew a bunch of money to start this museum, the Peabody Museum at Yale, and then so Marsh put a ton of different, new dinosaurs. Triceratops was one of the first, Triceratops, I think Allosaurus, Stegosaurus, a bunch of other dinosaurs Marsh described, and they're there at Yale. And then for a long time, nobody really did much dinosaur work at Yale until John Ostrom found Deinonychus back in the 1960s. And this is a dinosaur that kind of changed paleontology forever, dinosaur paleontology at least. This is the Velociraptor from Jurassic Park, is actually Deinonychus. And it changed the way we think about dinosaurs, where Deinonychus was sort of it was a challenge to the view that dinosaurs were all big, slow-moving, stupid, cold-blooded, antisocial, dull-colored, just boring animals. Because Deinonychus was small, like seemingly active, lithe, very bird-like, seemingly warm-blooded, very, very much like a bird. And so it really kind of challenged our view of dinosaurs and helped kickstart what we call the dinosaur renaissance from the 1970s through like the 1980s and 90s. But that's kind of the last time that Yale collected any new dinosaurs was like 60 years ago. It's mostly a historical collections there. They don't really have like a field program that I know of. They're not going out and finding new stuff, and so it's like, it's nice that they did this with Deinonychus, but it's also kind of a sign that like, they're not really doing much new field work. They're not finding new things, so they've got to go back to these specimens that were dug up 60 years ago.
And when you said Bone Wars, is that the same one that the book is based on?
Yes.
Okay.
Yeah.
I remember I was driving home one day from the lab, and I had you on my phone because it was your stream, and I'm just listening to you teach us, and then you told us about Bone Wars, and I remember I pulled over and I put the book in my Amazon cart that day because of you telling us about it. I have the book over there, and it's a
awesome.
wild story.
Yeah, isn't it? Yeah.
It's like some kind of like insane drama that's playing out of like the different the competitors going between them, and it was wild that's going on in at the time, and it was just, I don't know, it was a really cool
and that's
book.
where this museum got its start, was during the Bone Wars. There was this big rivalry between Edward Drinker Cope from the Academy of Natural Sciences in Philadelphia, and then Othniel Charles Marsh at the Yale Peabody Museum in Connecticut. That's where Yale is, right? Connecticut?
Yes. Yeah.
Yeah. Anyway, so like Marsh's wealthy uncle basically bought this museum for him, and he's like, here's your museum, and like that's how that museum got started, and that's the vast majority of their dinosaurs were found by Marsh's crews, and like the only real Deinonychus was a huge deal and changed dinosaur paleontology forever, but Yale has been kind of riding those laurels for like 60 years. Yeah.
Well, that takes them out. I thought they picked Deinonychus because it was like, well, it's known from Jurassic Park, and it's like this great. But I like
it's the only like well preserved small theropod dinosaur they have, because they haven't dug anything up in 60 years.
Gotcha. Well, I mean, I like the behind the scenes knowledge of it though. It's like kind of like
yeah.
the we picked it for this chivalrous reason isn't exactly the reason why. It ended up being
they picked it because that's what they have, and they don't got a lot else, honestly.
Yeah.
But it is very cool. It's very cool.
Yeah.
is one of the key moments in Yale paleontology history. It's really helped us reshape the way we think about what dinosaurs are, because it helped us solidify our understanding that birds are living dinosaurs. And so studying Deinonychus helps us get at that transition from extinct dinosaurs to living birds. By applying our SCORE and computer animation software, we were able to take the bones of Deinonychus from the museum drawer where they sit and reanimate them on our computer screens in 3D. And in doing that, we're able to see how this animal would have moved when it was alive, how it would have lifted its big, terrible claw off the ground while it was moving, and we've gotten a sense of how it might have used that claw in its terrible kick to kill animals as well. Now that we have the system built up,
the whole terrible kick thing, there are not very many paleontologists who still kind of cling to that old idea.
So what is the terrible click, Danny?
Well, so there's an idea that Deinonychus would have used that big toe claw to like rip open the bellies of other dinosaurs or like slash with it. You know, like kind of kick with that claw and just create these massive gashes with it. That is not how any modern animals use their claws, really. There's a newer idea that instead they used it like a hook for just kind of like hooking into their prey and standing on top of it and flapping their wings to stay upright.
As like modern
prey restraint.
Is like modern birds.
Yeah, like hawks and eagles do today. Yeah, it's behavior that we actually see, and they do cite that paper in this paper, which is nice, but I don't know. I wonder how much they really thought about it. Yeah, so it's like, I don't know. Yale has a reputation for being behind the times on stuff. I'll say it. And yeah, but anyway, not to take away from this is a really cool study, and as a methods paper, it's really neat. I'm getting nitpicky about like, you know, interpretations of different dinosaur behavior and stuff like that, and it's like that's not really what this paper is about.
But no, so I think that's important, right? Because I think, given my understanding of the paper, is that you're taking modern day bird movement, right, and you're superimposing how they move onto their bone structure, and you get a mathematical equation, and then you try to do the same thing with dinosaurs, but also working backwards, right? Like, here's the bone structure, you apply the mathematics to see how they could have moved. Using
the kind of articulation of the joints and how the legs would articulate and everything else, and like
like
spot on.
And like the connection points of like how much they have to connect, right? That all feeds in. You could, like, correct me if I'm wrong, Danny, but you can't use that to get anything about that hooking claw, because there's nothing today that does that kind of like jump attack like you were describing as like to rip open, right?
Yeah. In fact, it's kind of the opposite. We're like, we've got there's several different groups of birds that use their claws like this as a hook for hooking into prey, but nobody who like kicks their prey to death using their claws in the way that people have suggested for Deinonychus and the other dromaeosaurs, like Velociraptor.
I think
so. It's
like how you're saying, it's that what you put into this data set is what you're going to get out. So if your hypothesis is that it's that ripping motion, that can't be an output, because you can't model that and put it in.
Yeah, because no modern birds do that. Like, what are you going to use as your template there? I think that's what you're saying, right?
Yeah, exactly.
Yeah. So yeah.
Yeah, so I guess I don't think it's nitpicky at all. I think it's an important element to the study of like almost a limitation of it. Like, there might be behaviors that we hypothesize could happen, but we would never be able to yet use this if you're not finding a modern day bird of structural similarity that also does it.
Exactly. Yeah. And I do want to say also that one of the reasons why I wanted to pick this paper is I feel like it is it's a really beautiful illustration of how we work as dinosaur paleontologists, where we've got limited evidence, like we always talk about, Glenn, from the fossil record, and so we have to look for analogies, basically, from the modern world, from modern creatures that might be closely related to dinosaurs or might be dinosaurs themselves, like birds. And we're using the present as a key to the past. We're looking at how these modern birds behave and how their joints work and, like, you know, how they move and everything. And we're trying to apply that knowledge, which is easier to gain from modern birds. We're trying to apply that to dinosaurs to see if it makes sense and to try and like provide us a template for understanding them as once living animals. And like that's kind of a microcosm of just how dinosaur paleontology works. And I think it's a beautiful illustration of that, this paper.
Yeah. And
Together, we can rule the galaxy.
Hybrid Robotics, thank you for the resub. 3 months. Thank you
Hybrid
very much.
Robotics just subscribed for 3 months.
You got a piece of another raw science. We're getting dangerously close to finishing that out, y'all. Thank y'all
What
very
happens
much.
when that finishes, Belinda? Do you have to drink it?
Yeah, no. We do a raffle for a special glow-in-the-dark DNA pin.
Ooh.
Yeah, and then next month, actually,
That's awesome.
Lita's crocheting some of our SciComm characters, and so everyone who subs gets an entry,
Yeah.
and it's like, you know, tier 1 gets 1 ticket, tier 2 gets 2 tickets, tier 3 gets 6, just like with the Partner Plus. And so
Nice.
it's self-sub, but that way you get entries, and you could possibly win one of the custom plushies made by Lita. So we're just, you know, trying to pushing for that Partner Plus as an ambassador as well, and so it's, yeah. I thought it was fun because in the game Grounded, which we've been playing on Thursday, there's this raw science that you can collect in the game, and it's like this
Like we all have in our laboratories, just vats of raw science that a truck delivers every week. Yeah.
I mean, I wish, because apparently in the game it makes your intellect score go up, which is also kind of funny, and it's this pink goo. So I was like, well, yeah, let's have like a beaker and like put it up. So like each, like we started this month, and next month it'll be tier 1 is still going to be pink, but 2 and 3 will be different colors, and so then we'll see the science filling up, and I'm like, I think that's fun. I like that.
That's really cool. Yeah, I like that a lot.
And Smay was wondering about if there's any animal with like that one big claw.
Yeah. In fact, I can send you this paper real quick, Belinda, if you want to see it.
Absolutely.
Let's see, Fowler, predatory ecology of Deinonychus. But it turns out that there are several birds of prey today. Here's the link there in the chat for you. That do have a hypertrophy digit 2 claw. So just like the claw of Velociraptor or Deinonychus, birds of prey like eagles and hawks, their second claw, because the first claw is the one that points backwards, then they've got the second one, it's kind of one on the like inside, and then 3, and then 4. Their second claw is also the biggest. It's not super huge like it is in Deinonychus or Utahraptor or Velociraptor, but it's bigger than the rest of their claws. And you know, my old crew chief, this is Denver Fowler, the guy who was my mentor in Montana for a long time. He wanted to figure out how do modern creatures that have claws like this use them. And so he had to do nobody had ever actually studied that, even in modern birds. So his first paper, he called that his Claws 1 paper, was just looking at how do modern birds of prey use these claws. And like that had never been published before. So he looked at a bunch of different like taxidermied feet of different birds of prey and measured the curvature of the claws and the sharpness and the length and, you know, proportional sizes and everything else. And then he found a bunch of video footage of different birds of prey using these claws in different ways, and he kind of came up with like a schema for like, if you have toes of equal length, then you're doing this kind of thing, kind of like owls are. And owls have very long claws. Eagles tend to have claws that are they're shorter, but they're more robust, and they've got a really big digit 2 claw. And this is how they use it and they do the stability flapping and stuff like that. So it's like it's using the present as a key to the past, and using close relatives of dinosaurs like Deinonychus, you know, birds. It's using birds as like a clue to how these extinct animals would have behaved in a way that like it actually shows like it's drawing from real behaviors that real animals exhibit in nature, rather than like speculative ideas like, oh, kicking giant gashes in the side of a prey animal with claws. There's no animal today that does that. So, you know, let's look at what modern animals actually do for a clue for what these extinct animals would have done.
It's similar for the movement study, right? Like we're looking at the modern animals, how they move, and making an inference of it. And I love this figure because this is how you've been telling us, right? Like of some of these animals, like they'll clap on and they'll flap their wings, and it's just like it's really like a nice artistic representation to what might be happening.
My friend Nate did those illustrations. This is not AI art. This is a talented young scientist who made these, who did these illustrations here.
They're really,
And,
really pretty. It's a lost
yeah, isn't
art,
that great?
Danny. It's absolutely a lost
Yeah.
art of like being able,
Yeah.
like the scientific illustration world is even with like we talked about I think a few weeks ago. BioRender's I think adding worse to it as well because it's a nice tool for the clip art, but then you don't. It's another reason that you don't have to learn about some of these features.
Yep.
Yeah.
Yeah.
Should we continue on the video?
Yeah. Let's go for it.
Understand how to study how joints fit together. We can apply this to extinct animals from across the vertebrate family tree. So everything from how extinct animals would have flew to how they would have eaten to how other things would have run around. This is applicable to virtually any animal. It's extremely exciting to be able to take this data that's been sitting in our museum drawers for so long, to be able to harness it in a new way and to bring it into the 21st century and get it on our computer screens.
It's cool stuff.
This is
And,
my
and I love that she says that it's applicable to not just dinosaurs but a bunch of other creatures. Like that's why this is a methods paper, because it's like for dinosaurs like Deinonychus, there we can find relatives of these creatures living today, birds, and we can create a model for them using the range of motion and the joint articulation and stuff like that in birds. But if you want to do this for saber-toothed cats or for Columbian mammoths or something like that, you could look at their modern relatives. You could look at lions, or you could look at elephants, respectively, for those two groups, and determine the ranges of motion for those critters, and then try and figure out how would these extinct animals have moved. So it's really, really cool stuff. I love that this is like a framework for future research. It's like, here's an instruction manual. Other researchers run with this. See what you can do with it. It's exciting work.
It is really, really cool. I think it's kind of like how you were saying, it's like the data that you put in is the key part. So like even the other animals, like making the connection to what like the saber-toothed cat might be doing, you'd have to get that input of some really good videography and probably then like the skeletal structure of whatever you're comparing closest to, and then you can get it, which is fine. Like I think that's totally okay. And here's some of the other like supplementary videos that I just thought was also really beautiful.
I haven't seen these. Oh, these are cool.
Yeah, so like they zooming in on like the different bone structures too, and like the 3D reconstruction. Like this is, I think, really cool. Like where the surfaces
the articular surfaces there.
Yeah.
Where they actually come into contact. Yeah.
And like modeling that, and it's like a heat map on there too. And I was like, wow, this is like a really like good visual.
Isn't that neat? Yeah.
Yeah.
Very evocative. Like you just. Yeah, beautiful illustration of how this works. It just shows you visually. Don't tell,
So I think, Danny, one question I had, like, with this setup is how applicable is it to dinosaurs? And by that I mean, what kind of data do you need? Like, let's say we have the bird data, the modern day bird data, we have it banked. What kind of information do you need from the extinct dinosaurs? Like, how much bone do you need? How many samples do you need that you would be convinced by this? Like, how intact does it need to be? And
it's got to be, well, sample size doesn't really come into it too much, I don't think, because I don't think there'd be too much interest-specific variation on stuff like how your bones articulate.
Gotcha.
It seems like that's very basic nuts and bolts stuff, and it's not something I would expect to see a ton of variation in among individuals. But you do need fossils that are very well preserved, and when I say well preserved, I mean like you've just got beautiful intact bone in the field. It's not missing a bunch of chunks. It's not all scrungy. It's you could just nice and smooth. The articular surfaces should be nice and smooth. It should look like a bone that you would see today, and not something that's flattened like in a shale or something like that. Not something that's like all pockmarked and just like weirdly preserved, crumbling to dust. Like it's gonna have to be something that's like those Deinonychus fossils that they have at Yale. It's just beautiful and pristine in that sense. And the nice thing is that's not that hard to find.
Really? Okay. So when you were describing it, I was like, oh man, that seems like it would be.
Yeah, it's not as high a bar as you would imagine.
Okay.
Like in different geological formations, you get better or worse preservation of bone, and at certain like particular sites within a geologic formation. You might have really, really good preservation here, but it's bad there. And a lot of that comes down to like the original fossilization. So like if an animal dies and its flesh is scavenged or rots away, and those bones are just sitting there on the surface, and they're cracking in the sun and they're deteriorating, and then afterward it gets fossilized, that would not be a good candidate for this kind of thing. And we do get fossils like that, where like clearly they've been sitting out for a long time before they actually got buried. Yeah, but it's not uncommon to find really, really well preserved bone. In fact, a huge number of the dinosaurs that I've dug up have been just beautifully preserved like that. Like it's not a super rare thing. It's more common than you would imagine.
Nice.
Like it might be like 70% of the dinosaur fossils that I've dug up were well preserved enough where you could do this kind of work with them.
Oh, cool. Nice. And then you presumably need two parts of them, right?
Yeah. Yeah, you need articulating joints like
yeah.
that.
And does it
so like a femur, tibia, or
yeah,
radius, or excuse me, yeah, like radius, ulna, humerus, that kind of thing. Yeah.
Does it matter if they're from the same individual?
They should be from the same individual if you're going to look at the articulation.
Okay.
Because if you've got like a humerus from one animal and an ulna from another, it's like they're not going to be the correct size. Yeah, they need to be from the same individual.
I imagine the age will further pop into that. That could be a confounding variable, right?
I didn't even think about that. You're thinking about ontogeny. I've got you well trained, Bullen.
You got me going, Danny.
Yeah. I don't know how much of a role ontogeny would play with that. It's like, if you're an animal that's moving your limbs around, it doesn't really matter how old or young you are. Unless you're talking about like hatchling duck-billed dinosaurs, where they don't have the articular ends of their limb bones aren't fully developed yet, and they can't walk yet. It's like, yeah. But then even that would be a cool finding in and of itself. Like, yeah, these animals couldn't walk yet. And
yeah. And I guess too, like,
yeah.
independent of having different, bone specimens, like let's say you get them all from one individual, would it matter if that is a different age from what your data set is aged at? So like a Deinonychus that's much younger, does it move in any different way because of size? Maybe like maybe it's more built for speed at a certain point in life, or like the T-Rex, right? Like of what they hunted, right? The young T-Rex hunted something smaller, older ones bigger. Maybe there are differences in their gait behavior because of size.
I don't think we're at that point yet. Like these would be really, really cool follow-up studies, and like once this becomes an established method, yeah, and that's exciting to think about. But at this point, it seems like much more basic stuff than that. Like,
I'm just
like,
running
how
ahead.
far can they actually bend their knees and like stuff like that, which is cool. Like it's nice to be able to actually have a way to test these things. Like, what is the proper posture for an animal like this? That's kind of what I see this paper like giving to the world of dinosaur paleontology, is like, how do we create more accurate representations of these animals? What's the proper posture for their limbs? What should be like the resting stance for where their knee articulates between the femur and the tibia, like that kind of thing, if that makes sense.
Basically, I'm jumping ahead to assuming we have all this knowledge already built in from like the fundamentals.
Yeah.
I get
Which
excited.
is cool. Yeah.
Even like this is another one that they had. They zoomed in of like that, like where the claw was looking. And again, like out in the video, they were showing like what the movement would be. And I liked this supplementary figure, I think, the best because they had it from multiple angles.
Oh, that's neat.
Right, like they could show you from the over side head. I think like that's a really valuable tool to be able to reconstruct everything in 3D space like that and really show off.
Yeah.
Just how the organisms have that gait and walking behavior.
And it's cool because we don't have any modern animals that have got their second toe lifted up like that. So like figuring this out that's pretty neat, and this would, like, I hope that every like studio that does CGI work, Industrial Light and Magic and Framestore and all these other groups like that do CGI for movies and documentaries and stuff, I hope they are paying close attention to this paper, and I hope they're very excited about it, because this is very relevant to what they do, and it gets into like educating the public and creating more authentic dinosaurs for public consumption in documentaries and films and stuff like that. Like this all goes into that, and I think that's really neat.
Yeah, I worry about the the benefit for public consumption, because they don't seem to be that willing for something like that, right? But for public engagement. But there was it was on Apple TV Plus, that really good documentary series,
Prehistoric Planet.
That one, yeah. I feel like they would really appreciate something like this, and really
Definitely.
Yeah,
Yeah.
push hard to have something like this, like the accurate representation of what's being
Because
done.
Stuff like that is really important. Like, even we were talking about this on my stream a couple weeks ago, I think, maybe last week. But Blint, you've seen The Empire Strikes Back, right?
Yeah.
Yeah, and the Hoth battle scene at the beginning with the big AT-AT walkers and everything.
Yeah.
So that was done with like stop motion animation and stuff. And the Phil Tippett, who I think was in charge of the stop motion animation, he had his team go to a spot that had elephants, and they painted these lines on the elephant's legs, and then took video of them walking, and so they could see how the legs articulated and everything, because they're just trying to get a sense for like a big four-legged thing. How can we make like something like an AT-AT walker? How can we make it walk across the screen and look convincing? How can we make it, just look cool, but also realistic enough where people like get invested in it and believe it?
Yeah.
It's not something crazy jumping across the screen that takes you out of the movie and destroys your immersion. They wanted to do some so they looked at elephants for that, and that's even in like something that's fantasy, like AT-AT walkers aren't real, as much as that would be cool if they were. But like they're still drawing from the natural world in order to create that. These sorts of studies like this is going to have, I think, really important ramifications for people who want to reconstruct dinosaurs in films and documentaries and video games and stuff like that. It's cool to think about.
Yeah, absolutely. And
Bauxite, nice.
Raiders, welcome in. Danny, you always know B4UX's name much better than I. I never came able to pronounce it. I was like, oh, B4UX.
Yeah,
You said...
right?
Yeah. You always get it right. I'm just like, oh, I never know. Raiders, welcome on in. How was your stream? See, Hanny, I see that word and I don't know how to pronounce it. Even like without the numbers now. How was the stream? Welcome on in, y'all. I know it's B4 is one of our game dev friends. Go drop them a follow, y'all. How was your stream, B4? Tell us all the things. For the Raiders coming on in, my name is Balint. 1/3 of science streams and 2/3 are my wife Lita and our daughter baby Alona. We're research scientists and we do interactive science streams here on Twitch 7 days a week. We're joined today by Professor Danny Anduza, single greatest science communicator on the planet. And we're chatting fossil science, genetics, and both of us picked methods papers to chat about today, which is always a good time. It was great. We were building a ham control radio software but using a game engine, Godot. Very cool, B4. Excellent.
Nice.
I think we got a couple of friends in the community that use the Godot game engine. A lot of Unity folk and one or two of the Unreal game developers as well. B4, thank you for the raid. If there's anything you want to show, feel free to drop it in the chat. We'll show it off.
I like that you call him B4. It's getting back to Star Wars. It sounds like I don't know.
The droid.
B4.
Yeah.
Shut down all the trash compactors on the detention level.
I know Alex wrote it out like bauxite, but it's one of those things that's now so ingrained in me. I'm just like, it's always just B4UX. And
Yep.
it's instead of the elite letters throw me off so much. Drop no worries. I hope the kiddo feels better soon. Thank you, B4, for the raid. Or bauxite. Appreciate you very much. I hope the kiddo feels better soon. I mean, I see it. The 4 is the A, and it just leaves.
It's charming though when you say B4. I like that. Yeah.
Thank you, Danny.
Yeah.
But yeah, so I thought this was a really, really cool paper. I think it's very much in line with how we've talked about of like looking at your modern day animals to inform things about the past. Do you see like what was the coolest paper figure in here for you, or like what was the thing that got you really excited?
That's a good question. I think figure 3 I thought was particularly interesting. Like just looking at these different toe bones there in the foot, and I don't know. I guess this is what we were also looking at with that animation there, but it's just really neat stuff, especially because this is not a perfect analog to like any modern birds, or no modern birds have got that raised digit 2 claw, and I've seen like different orientations suggested for this claw. There have been some people who have suggested that the claw actually like would have contacted the ground, that it wasn't actually held upright. And here they're saying, well, it makes sense for it to be held upright, and that actually matches with some fossil footprints evidence as well. We have trackways of dinosaurs like this. There's one in Utah that I saw this past summer, and I think it was the first ever dromaeosaur trackway, where there's just, you know, just side by side, just two toes, because the third toe, or the second toe, is raised like that. And so it's cool that we've got like independent confirmation from different lines of evidence there. And that's what these animals were actually doing.
I guess that was one question I had for you, is how do you know where to position that giant claw? Right, when you're doing this, because it almost feels like it's one of those where your model will fit depending on how you arrange this. Right, so it's cool to hear there's like other like evidence of like the footprints of how they could have walked, and that's feeding into this model. But just from like, if you didn't have that and you're trying to use this method when you're assembling the bone structure, how do you know that it's going to be upright versus a claw that's pointed down?
Right. Yeah. For one part, it's the shape of the claw itself. Mm-hmm. Like, for claws, we call them unguals. That's like I keep saying claw, but like I really want to say ungual because that's the term that we use. Unguals are that's like the bony core of the claw. And so unguals that are actually contacting the ground don't have a super sharp curve to them. They're going to be a little bit more flat. And so you look at the two toes right there that are contacting the ground in that figure. Mm-hmm. If you scroll down just a little bit, check. There we go, yeah. Those two that are contacting the ground, the unguals are a lot more flat, and then you've got the big killing claw, the hypertrophy digit 2 claw, that's much more curved. So that's clearly like not resting on the ground or anything. But like how much of the actual toe there was contacting the ground, like that's been a mystery for a long time.
Yeah.
And this paper helps elucidate that a little bit too, which is cool.
Is it... Yeah. If it were really curved, like if it's this curved, if it were on the ground, would you have the likelihood of actually impaling yourself as well?
Maybe, or it just, I don't know, it's like it wouldn't do much good being on the ground like that, because it would just be like the tip that's contacting.
Yeah.
And then it might break off, or like...
So there's like
it seems like the reason why this claw evolved to be, you know, held up like that is to keep it sharp. You know, almost think of it like the retractable claws like in a cat or something. Cool. Like this is a different way of retracting that claw so it can stay really sharp and it's not contacting the ground all the time and getting dulled. If that makes sense.
I like that. It's like the functional feature of it too, like going into it. It's like not just like it wouldn't help with balance, let's say, or like the function of your feet, but also like then what would be the point of having something like this? And so added together, that's why we say probably upright, and then you build it into the model like this, and it's still like the walking motion makes sense.
Yep, and then you compare that to like fossil trackway evidence, and it's like, oh yeah, it definitely matches, and that's pretty cool.
That is really cool. And I'm glad that they picked the Deinonychus. It's like I know it's like the classic one you were saying.
They had to, but like they're lucky that Yale was the right place to do this kind of a study. I'll say that because they've got Deinonychus there. Yeah.
Well, I think it's also like you were saying, it's almost like the easiest way of having something like this work if you have the complete skeletal structure, right? So that like in really good holotypes, and you're just, if it works with this, like this is how you build the system, then maybe you can work backwards and have like less complete skeletal structure, versus like if you started with the less complete, then your model might not be good.
You don't even need a complete skeleton for this. You just need limb bones that articulate, you know? So like you could just have a femur and a tibia, and you should be able to do this kind of work as long as they're well preserved, you know? You know, like you might not know what the rest of the animal looks like, but you can at least know, you know, what its joints look like and what the range of motion is and all that. Yeah.
Oh, okay. So I guess it's like because they also, in addition to knowing how to interact, they were able to model how the animal walked, and I guess for that,
Yeah.
you need much
For that, you
more
need
than yeah.
For sure. Yeah.
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 It's super cool, and I like too how you said how they said in the video is that there's the possibility of pushing this towards other animals as well, and how they might be walking too. Like one of my favorite figure was actually was this one too, but I really like the heat maps of like where the bones touched and
The
interacted.
articulation score there.
Yeah. I thought that was a really clever way of arguing too of when you're assembling the dinosaur bones. Like it almost feels like you can use this as a proxy for does your assembly make sense?
Yes. Yeah. And that's been an issue before. Like, for a long time. Here, let me see if I can find you an image. Let's see. AMNH. Tyrannosaurus. Upright. Yeah, there we go. Let's see if I can open this in a new tab and then grab the link. That's not going to work. Hang on. That should work there. Yeah. So, classic image. There's the link in the chat. The cover of Jurassic Park, the novel, and like the Jurassic Park logo, that's based on this Tyrannosaurus specimen, how it used to be mounted in the American Museum of Natural History in New York. This is totally incorrect. These animals did not walk upright like Godzilla or something. You know, they didn't drag their tails on the ground. And in order to do this, several of the bones in the tail actually had to be broken in order to get it to assume this pose. And so this was an old example of like, well, you know, dinosaurs are reptiles, and reptiles are slow-moving and cold-blooded, and they drag their tails on the ground.
Mm-hmm.
They were following that assumption rather than actually following the way the bones articulate with one another. So like this sort of thing, you would not do this today for a number of reasons, but like one of them is that you've got studies like this now that basically you can quantify how those bones should articulate, and you would realize that like this is not the proper pose for this animal. Does that make sense?
It's surprising too that they went in and broke the bones, right? Like that almost, I feel like that's not just a misassembly, that's forcing a predisposed like hypothesis, and you're forcing it into that.
Yep. But this is just how it was done back then. There's a place in Belgium where a bunch of iguanodon skeletons were found. Here, I'll find you an image for that. And this is like, nobody had ever found a complete dinosaur skeleton until this moment. This is in the late 1800s. Yeah, here. Let's find the Bernassart iguanodon. That'll work. Yeah. Oh, this one's even better. Beautiful. But for the first time, like, we actually had complete skeletons of of dinosaurs. And the paleontologist, he actually wasn't trained as a paleontologist. Louis Dolo was, he may have been an engineer by training, but he was told to like put these animals together, and first time anybody ever had complete skeletons of dinosaurs. And so he looked at some modern animals to try and figure it out. He discovered, he's like, oh, iguanodon, it should probably walk on two legs. And he looked at emus and wallabies as kind of his like his inspiration for how the animal should be posed. He's like, if you're going to stand on two legs, then like a wallaby, maybe you've got that big thick tail to kind of rest on like this. And so this is where the broken things, you can see where the tail kind of kinks about a quarter of the way down.
Yeah.
And there are several broken bones in there. Like, he had to break some of the bones in order to get that to work. But
do we know what the logic was behind that? Like, it almost seems
like,
counterintuitive. Like, if you have to break the bones.
It was. They knew so little back then, and this was already cutting edge stuff, because it's like, he's got the limbs in the correct position, at least, where it's like they're directly under the body.
Mm-hmm.
The animal's got an upright stance, but it's not. I guess it was like it was like, this is an animal that nobody's ever seen before, and we're gonna use some modern animals to try and figure it out. And like wallaby is what he hit upon. He's like, well, wallabies bend their tail like this. Maybe these animals could do the same thing. When I say break the bones, it's like, it's not like they had to physically break them, but they had to like disarticulate them. They had to dislocate some of those tail vertebrae in order to get that tail to curve in that really unnatural way.
Gotcha. And then the software that we're looking at in this paper today would account for the fact that there's no connection point between those.
Exactly. That's the point I was trying to make here. Thank you. Yes. So yeah. So this is one of the ways that dinosaur paleontology has advanced since the late 19th century.
It's really cool. Like, and I was just my naive thought of like, as I was reading the paper, I was like, oh, I wonder if this could have been could be used today to kind of predict the proper structure. But it sounds like we've come far enough along where we wouldn't have needed this software. But maybe it's like
we're hopefully not making grievous mistakes like this anymore.
Yeah, but maybe it's like a secondary confirmation.
Yeah, and a way to. One of the things I'm interested in is like maybe this might have some application for like the wiggly waggliness of dinosaur tails. Like that's something that my old boss Jack Horner used to talk about. It's like, oh, dinosaurs in movies, their tails are too wiggly. Like they'd be much stiffer in real life. And you know, if we could use the methods from this paper and apply them to dinosaur tails, maybe you could tell how flexible the tails should actually be. Because it's like it's difficult to actually do that in real life with the bones, like moving them around in physical space. But if you can scan all of them and put them into a computer and then run them through this program where you actually like, you know, you get numbers on like where's the best articulation points and everything, that could potentially give you much better insight for that kind of thing.
Because there is the hypothesis, right, that they were the way they were walking, and actually there's this image from Charlie's Dragons about how it's supposed to look, right? The comparison of the T-Rex.
Oh yeah, that's Stu, the T-Rex. Yeah, so the tail is held up for balance behind it, and like this actually makes sense for the animal's anatomy. Like,
and how they could use, so like if we could model like the flexibility of the tail, isn't there a hypothesis that they could direct, like if they were fast moving, it's almost like a rudder?
Sort of, yeah. That's actually been suggested for Deinonychus. Yeah.
So like for the like how much of a range of balance do you have is what it for me, the layperson on this, feels like you could also then use it for.
Yeah, for sure. And another thing with dinosaur tails is that they've got these on the caudal vertebrae, the tail vertebrae, you've got these processes that come out to the sides. They're like little wings that come out to the sides of the vertebral centrum. They're called transverse processes. And those are attachment points for muscles that run down to the back of the thigh. So there's like a lump on the back of the thigh bone, back of the femur. That's an attachment point for muscles that run from that to those processes on the tail. This big caudofemoralis muscle. And this is different from how modern birds work. Modern birds do not have long bony tails. And they don't have a great big caudofemoralis muscle that goes from their thigh to their tailbones like that, like dinosaurs did. And so this paper might offer some insight into how to like properly reconstruct that. And the tail moving back and forth side to side, that's probably part of the dinosaur walk cycle. It's like as these animals are moving, the thigh bone is swinging backward and forward, and that's because that muscle that goes from the thigh bone to the tail is like contracting and uncontracting. There's a lot going on there, but like there's a lot to unpack. But hopefully this paper and the methods within it can be used for like for further elucidating some of these things that we don't know a whole lot about right now. Yeah.
I think it's really cool that there's that muscle that's no longer there in your modern birds. Like they lost the tail, the muscle, it makes sense, is gone. I wonder if that's like the rebirthifying problem too, of like having that muscle like regrow and reconnect could also be an issue.
Have you seen the video, Balint, of the chicken walking like a dinosaur? They basically stuck like a plunger to its butt to like simulate what a long
I have
bony tail
not.
would be
I don't
like.
think I've seen this.
I've got it right here. I'm putting it in the chat for you. There. Yeah. This is some quality science here.
All right,
That's a regular
control.
chicken.
Excellent. Yes.
And the way that the chicken moves actually changes. Like, it changes
Huh.
its posture, it changes its gait a little bit. And this is just having a prosthesis on the back. There's no muscle attachment or no caudofemoralis action or anything. It's just having a little bit of a counterbalance, you know, on the back of the animal like that. But it kind of changes the way that it walks, which is really interesting.
And I wonder for how long the animal had this. Was it just put on video recorded, or was there a time to adapt to it, and how long that adaptation takes? And
I'd never even thought about that. That's a great question. I don't know. Yeah.
Moonrise says cheetahs have a plume of fur on the tips of their tails that act as a rudder. Would these Maniraptorans have something similar with feathers?
Probably Moonrise Rabbit, yeah. We do have some evidence that other dinosaurs, like Oviraptorosaurs, had like a big plume of feathers on the back, like a fan of feathers. Usually when paleo artists are reconstructing dromaeosaurs and troodontids, they usually put a similar kind of plume back there as well. But we don't know if they had that necessarily. Yeah, it's a good question. Good question. But with Deinonychus, you know, this dinosaur from Yale, it's got this long, stiff, bony tail, and that was like one of the first pieces of evidence that, like, shoot, dinosaurs didn't drag their tails. Because this thing, like, it's like a broom handle coming off the back of this animal. It's very rigid. Like, that's not a flexible thing that's just dragging on the ground. Oh, nice, Sculpin.
Sculpin, thank you for those 100 biddies. Appreciate you.
So, like, Deinonychus is really important in that respect, but John Ostrom, you know, there's video of him talking about this, actually. Should I see if I can find that real quick for you, Bullet, and timestamp it?
If you don't mind, Danny.
Yeah. PBS The Dinosaurs. Ostrom, let's see if that pulls it up. There we go. Okay. I've got it here now. I just got to find the right timestamp. But, you've maybe even seen this before on my stream, Bullet, but I love the joy in Ostrom's eyes as he talks about this. Here we go. And timestamp. Copy. There you go. Yeah. So this is the guy who discovered Deinonychus back in the 60s.
I have seen this video, yeah.
Later.
Yeah. But he talks a little bit about the tail at the end of it.
We watched this video recently, right?
Probably.
Not too long.
It comes up a lot.
No, I just remembering it's like there was one recently about, we were chatting about like inaccurate dinosaur statues as well, and I think we were...
Yeah.
Right.
They kind of use that for a framing device there. That mural is still there at Yale's Peabody Museum. They're redoing the whole fossil hall, but that mural is such a piece of paleo art history that they're leaving it up. It's a fresco rather than a mural. It's actually painted on plaster using like egg tempera paints in the way that like the ancient Greeks did.
And I'm assuming it's outdated at this point, right?
Extraordinarily outdated. Arguably it was outdated for the 40s, but it was painted during World War II. Yeah, and it's supposedly one of the largest paintings in the entire world.
Wow.
But yeah, check it out.
I'd like to introduce you to the largest painting in the world. It portrays time over a span of about 350 million years, from the Devonian period back in the Paleozoic, all the way up to the end of the dinosaur era, the end of the Mesozoic. It's called the Age of Reptiles.
Reptiles, that's a pronunciation that's now extinct. Transatlantic, like mid-century. Reptiles.
Here is the old picture of dinosaurs. Pea-brained giants lumbering through lush tropical foliage, gorging themselves on leaves and grass. For years, the stereotype was magnificently represented by this mural.
Since the 40s, when it was painted, a number of discoveries have been made, and new ideas have been thrown into the debate. And perhaps one of the most important discoveries was made by me.
I love the joy in his voice and his face there. Oh, it makes me so happy. Yeah.
It was 1964. John Ostrom recalls that he had been digging for months in Low County, Montana, and not finding very much.
Yeah. He's going to talk about the tail in a little bit. Yeah.
His crew had packed up their equipment and were heading for their cars.
You know, we've been looking for 5 years before we found anything as exciting as this.
Close to where the cars were parked, Ostrom noticed something in the rock. Since his tools were already stowed away, he began to scrape the dirt with his fingernails. This is what he saw first.
Yeah.
Startling. I couldn't believe what I was looking at. Notice the extraordinarily large claws, sharp, curved, clearly the hands of a predaceous animal. Associated, found very close to this, was this object, obviously a claw, which I thought belonged to the hand because it looks very much like the claws on those fingers. But there was no place for it to fit.
Yep.
And I puzzled over that for some time. But we found the answer.
Yeah.
Turns out that that claw didn't belong to the hand at all. In fact, it belonged to the foot. And something more came to light in our quarry. And show you just a part of it. Bits and pieces of the tail, encased in bundles of ossified tendons all along each right side, left side,
Those are actually like
underneath.
ligaments that turn
This
to
made
bone
the
over the
tail
course of the animal's
completely
life.
stiff, like a
So you can see the end of, because of like how it's fossilized. Like, is that's part
Yeah.
presumably like, is that rare for us to get tendons?
No. In fact, actually, loose bits of tendon like that are maybe the most common piece of dinosaur that you find in the Hell Creek Formation. They'd either be from Triceratops or from Edmontosaurus. But you just find this stuff all over the place. Just eroding out of hillsides and stuff. Because it's like a really common part of the animal. These bundles of ligaments and tendons. I think they're actually ligaments. I think ossified tendon might be a misnomer. But they're ligament that literally turns to bone over the course of the animal's life, which is really cool to think about.
Yeah.
So, like, it wouldn't be bone earlier in the animal's life, but later on, as the animal matures, and like as it needs more rigidity in its skeleton like that stuff ossifies.
Gotcha. So it's ossifying during the lifetime of the animal, so it's bone by the time it dies, so then it's preserved the same way as regular bone. It's just, you can tell the difference between what was once tendon versus what originated as bone.
Yeah, because it's like, it'll be around the other bones in weird ways that you wouldn't expect bone. It's like, it's clear that this was tendon that later turned to bone.
That's really, really cool to be able to see like that much of like, look into the life of the animal of how something like that has changed.
Yeah, and I don't know if we have examples of that in modern creatures. Like, it might... I don't think there are mammals that do that. I could be wrong, but I don't think so. It's just kind of like a cool dinosaur thing.
Yeah.
That's,
do birds do that?
Yeah. I don't think they do, but birds today are weird because they've got super modified skeletons for flight, so they like want to reduce the amount of bone as much as they can.
Right, it's like that
so
weight
not
decrease.
Be doing that kind of thing.
Yeah.
Exactly.
So you'd want to have lost that feature of it. Yep.
Exactly.
So your only, I guess your only look would be like emus or ostriches, maybe the flightless large birds, and see if it's maintained there or not.
And they are secondarily flightless too. Like, their ancestors could fly, and so
then probably it's gone there too.
Exactly. Yeah.
It's really it's so cool though.
Isn't this neat?
Yeah.
He's about to talk about the purpose of the. He says a balancing rod or something for the tail. Yeah.
what I picture as a balancing pole. It kept the animal, helped the animal keep its balance while it was using those sickles on its feet for killing whatever it was hungry for.
So John Ostrom proposed that slashing idea
Ostrom
way back in the
named
60s.
the animal Deinonychus.
just because of how it was posed here, right? Presumably, like
Yeah,
it's kind of
yeah.
leading to that jumping behavior, like you were saying, and that like slicing open
Yep.
as well.
Yeah,
It's-
yeah.
I like all the evidence that adds up to how it is posed. Like you were saying, the walking, what ends
Uh-huh.
up like actually looking like with the animal, and then like I really like this video. I want to, if you don't mind, Danny, I want to show this again of just like the different posturing
Yeah.
that they showed us, and like again how everything articulates and fits together. It's just it's really elegant. When I didn't realize like so many things are coming together to make
yeah.
this point. Like it's not just that they just fit together, you know, what modern birds do and then like superimpose it, but there's so many lines of evidence for what Deinonychus did
Yep.
that it's like a further confirmation. It's I guess it's the methods paper in the same way that the other one was, where it's like we have known things almost as much known as it can, and this validates it, so therefore it's a method that we can use on other things.
Exactly. It's like it's proving the efficacy of that method because the method works for predicting things that we already know when you apply it to that, so we can probably trust it for stuff that we don't know as well.
Yeah.
And that's the beauty of a good scientific method is you can test it against like, well, we already know this thing. Apply that method to it, and if it gets the wrong answer, throw out that method. It's no good. But if it gets it right, then we're cooking.
Yeah.
So,
Which is it's and this one too, right? The multi-angular one is like, again, I would love to see the footprints superimposed on this. Just seeing like how well that matches. I'm sure it could be done, or maybe they even, you know, did it at some point, just didn't make it in the paper, but like as a final confirmation on it almost of how it works.
Mm-hmm.
And
Cool stuff.
yeah, Nelf was wondering, are there any modern birds whose ancestors also never flew?
No. All of the birds that we have today had volant ancestors. They evolved from ancestors that could fly. Yeah. It seems like only flying birds are the ones that made it through the extinction event at the end of the Cretaceous. All the birds that couldn't fly died out and left no descendants. So all the birds that we have today are descended from birds that originally could fly.
And when you've told us that before, Danny, if I'm right, like the radiation of all those birds, the actual amount that survived is a really small percentage of all the ones that were there at the time.
Yeah, it's like single digits. It's like less than 10% of all bird species that existed at the end of the Cretaceous also existed after the extinction event. It's like very few made it through. There's actually a really interesting paper, a couple of papers about that from the past couple weeks that have been making the rounds on the internet. Last Tuesday when I was at the museum listening to some talks, you know, I overheard some of my colleagues talking. I'm like, oh yeah, is this that bird phylogenies paper? And they're like, yeah. There is one paper that said that like, oh, all the modern groups of birds evolved way before the extinction event, and then, you know, made it through. And then the conventional wisdom is that there's a massive adaptive radiation after the extinction event. And yeah, so I don't know. There's some controversy about that now, but it seems like that consensus view is probably going to hold for now. That like almost everybody died out during the extinction, and that massive radiation of modern birds happened after the extinction event. Those few species that survived then multiplied into the over 9,000 species of birds that we have today.
It's like they had the niche that they could expand in.
Exactly. Like, you know, nature abhors a vacuum. There is a vacuum there, and they expanded to fill it.
Yeah, I mean that's the beauty of specialization, right? And that's,
Uh-huh.
I mean, it's really cool that ended up evolving too. Oh, and Alex linked us to this video, Danny, with the further explanation of the chicken tail.
Uh-huh.
The team from UIC won the Ig Nobel Biology Prize in 2015.
Oh boy.
The Ig Nobel frustrates me to no end, because I feel like it does so much damage to the scientists.
It's just saying, oh, this research is silly or stupid. It's like, no. Yeah.
Yeah. I'll
This chicken with
I'll link it, Alex, into the about section too. I don't want to give this guy too much credibility if he's talking about the Ig Nobel. Even if it's like video about the data, it's just because like so many people have used the Ig Nobel as like reasons to get rid of research funding, because it's like, look at how quote unquote stupid like this is, and yeah, it's frustrating on that front. We did have a quick question, Danny, on dodo birds, as they were also flightless. Is that something that, like, they presumably lost that flight ability?
Oh yeah.
Yeah.
So dodos are a kind of pigeon, and so their ancestors flew to the island of Mauritius. That's how they got there in the first place. And then things were so good there that, like, over, you know, thousands of generations, dodos have been around for like 10 million years before they went extinct, something like that. And they, like, survived all of these, like, volcanic eruptions and famines and droughts and, like, all kinds of other stuff. And it was only the arrival of people who killed them all, largely through the introduction of critters like rats, that dodos went extinct. But they were supremely well adapted to their environment. But they were able to get really big and lose the ability to fly because they didn't need to fly on this island with no predators, you know? And so, yeah, but their ancestors flew there in the first place. That's how they arrived.
And there was no, if I'm correct, like, no predator reaction as well. Like, they didn't know that they were being preyed, so there was no fear response because there was nothing to fear.
Exactly. Yeah. Yeah. Which makes a lot of sense.
Yeah.
But yeah. So the ancestors of the dodo could fly. They looked like pigeons. Dodos are part of the pigeon family. Yeah.
Which is not just like in the movie Ice Age, right? Where
that really bothered me, that depiction of dodos. Like, they were not stupid animals. They also didn't exist in North America. They're specifically from the island of Mauritius, you know? It's like they might as well have put kangaroos, you know, hopping around in Yellowstone National Park or something like that. Like, it's that level of ridiculous.
Gotcha. So they're totally in the wrong zone of everything.
Exactly. Yeah.
Yeah. Danny, how it is getting late, my friend. I want to make sure that we're not, like, you have dinner to go to.
I should probably go pretty soon. Yeah.
I just want to make, like, because we usually round out even a little bit earlier sometimes. I just want to make sure I'm not keeping you too late from the lasagna tonight.
I appreciate that. I am looking forward to heating up that lasagna and having that for dinner. So let's do maybe a few more questions and then, like, wrap up the paper or something. Yeah.
Okay. So y'all feel free to ask. I know Alex said there were 3 experimental groups: the normal chickens with the tails, and a group with a weight to simulate the extra tail weight. And the chickens were raised using the tails and the weight, so they would have adapted throughout.
Interesting. Cool.
I think that's a nice important control to be able to have, because if you just stick on the weight, like for the chicken tail experiment, I can see there being adaptation periods or the musculature forming. And even though that muscle is gone, I can still, if it's a lifetime, they're going to be adapting differently. And thank y'all very much for the follows. Pero, Loco Yo, and Iyakia Yaka. Welcome on in.
And I see Dame Karen say, it's a kid's film, Danny, not a documentary. Well, but that's the thing, is that Ice Age is really, really good in certain ways. Like, as a paleontologist watching it, like, they've got all of these different Pleistocene creatures that they introduce, and it's like, somebody in the production of this film really was enthusiastic about Pleistocene mammals. You've got critters like Macrokenia, you've got Glyptodonts, and you've got ground sloths and stuff like that. It's like, you know, there were some smart people who were involved with this, who were passionate about Ice Age animals, and then they throw in this stupid plotline about the dodos and all that. It's just like a funny thing for, you know, for laughs, and it like hammers home this idea. The whole point of the dodo thing is like, oh, dodos are stupid, and they were destined for extinction, and it really, really rankles me because they were not destined for extinction. Like, these were beautiful, extremely well-adapted animals for the environment that they lived in out on this island. It's almost like absolving us as human beings for the crime of driving these creatures into extinction, you know?
Like, they were already so stupid they were going to die anyway, independent of us doing anything.
Exactly, and that is not the case. They had survived on that island for, 10 million years after having first arrived there, and they had adapted really beautifully to that. And, like, I think, it really does dodos a disservice, Like, it turns them into a punchline in a way that they do not deserve. We already we literally drove them into extinction, and now we're, sitting and laughing on their grave and telling jokes. It's like very disrespectful.
I haven't ever heard it framed that way, and I'm glad that you framed it that way, because that makes it all the more uncomfortable, right? Especially, and I didn't even realize in watching the movie, it didn't even register to me that it wasn't even like geographically making sense.
Yeah.
And you're right, like a lot of other stuff they did get accurate in terms of like what animals were around, and so it can be like, obviously you're not supposed to take it as a documentary, but it could be misleading. And speaking of misleading, so KK Gatagonia was asking about, have you seen The Land Before Time? And what is your feeling
when I
on
was a kid? I don't remember anything from it.
if there was like a strong inaccuracy or accuracy behind that one.
If I remember correctly, The Land Before Time makes a mistake that a lot of like popular dinosaur media makes, where it kind of just throws together all these different dinosaurs from different times into the same time. So like I think there are like Stegosaurus and Triceratops living side by side in that film.
Yeah.
There is more time separating Stegosaurus from Triceratops than separating Triceratops from us. Like, Triceratops is closer in time to Twitch.tv than it is to Stegosaurus. So, like, those are two very popular dinosaurs, and that's why they got thrown in together in that movie, but that is not at all, how things actually were in life. There was no, like, single time of the dinosaurs, Land Before Time. Well, time existed back then, too, and there was a lot of it during the age of the dinosaurs. Dinosaurs were around for about 160 million years, and, like, you've just got this constantly changing cast of characters all the time, where new dinosaurs are evolving and certain groups are going extinct as conditions change, and it's just this it would be kind of like having a movie about American history, and you have George Washington and Abraham Lincoln and I don't know, Arnold Schwarzenegger all like going to the ice cream shop together and then playing foosball. It's like, okay, that's fun, but that's like, they did not live at the same time, Yeah.
I'm not me, I'm you. Did you ever see, Danny, this other Disney movie? It's like Dinosaur. It was a CG animated one. It had the same plot as Land Before Time.
They did the same thing.
Was that also the wrong dinosaurs were mixed together?
So the dinosaurs that were in that like are pretty well rendered and like very clearly like lovingly rendered, where like they put a lot of attention into actually having specific dinosaurs that are like, they look like the real thing, but also in that you've got dinosaurs from different times stuck together. You've got Brachiosaurus and Styracosaurus hanging out together and stuff. Carnotaurus, which is from the very end of the Cretaceous period, and Iguanodon, which is from the beginning of the Cretaceous period. There's like 70 million years separating them, and but they're fighting, stuff like that.
What movie, or even I guess game, would you recommend if people wanted to see like what's actually accurate in terms of time?
Saurian is a really, really good game, and I played that on stream a number of times. I should do it again sometime soon. It's been like years since I played a video game on Twitch.
I remember last year, because you had the green screen that came down, and
was that last year? I thought it was. It might have been. It might have been 2022 or 2021, but anyway, yeah.
I was around, Danny. I remember. I downloaded it because of you.
Oh, you definitely were. Yeah, for sure, for sure. But that game, it's really cool in that you play as a dinosaur, as a dromaeosaur, like Deinonychus, but it's set in the Hell Creek Formation. And so all the dinosaurs that you interact with are other dinosaurs that lived in that same time and that same place. And the Hell Creek is really cool because you've got a lot of the most famous dinosaurs in it. You've got Triceratops, and you've got Tyrannosaurus, Pachycephalosaurus, and Ankylosaurus. And all of those dinosaurs were actually neighbors. They all lived together. And that game does a wonderful job of not just showing them together, but also showing what the whole environment would have looked like. Including for everything from the climate to the plants. Oh man, the detail in the plants is exquisite. To to the non-dinosaur creatures they lived with, various species of turtles and champsosaurs and mammals and stuff. It's really excellent. Like, if you want a picture of what the Hell Creek Formation would have looked like, what are the dinosaurs that were palling around with T-Rex? Check out Saurian. It's an excellent game, everybody. Yeah.
And I think you did a review on it, right? There was like a game review that you had on your YouTube.
That was Prehistoric Kingdom, but yeah.
Oh, that was gotcha. It was
which is like that's like a Zoo Tycoon type game, except the dinosaurs and other extinct creatures in it are so well done. So like, there's like a fantasy element into it where it's like, yeah, extinct animals in a zoo today with people walking around. But the way they do it is beautiful and very thoughtful, and I like it a lot.
Like versus the Jurassic World or Evolution, right? That I think you
Which
reviewed
is a lot
that
more
too.
Like fantastical and like, yeah, there's a lot of like hybrid monster dinosaurs and stuff like that. They're kind of like hobbled by, they need to make their dinosaurs look like the ones from the Jurassic World movies.
Yeah,
which tend to be pretty lousy. So that's kind of the millstone around that game's neck, I guess.
Yeah. But I think it's cool to have the accurate ones. Like, we've been playing Grounded, which is like Honey, I Shrunk the Kids. You get shrunken down, you're in a backyard and like looking at the insects, because it's all like different insects that are coming after you, and like for the most part they get the morphologies correct, and even the swarming behavior, like the ants that are patrolling won't bother you, but if you hit one, there's a recruitment pheromone and others will come in and for you, versus if you go in the colony, they'll actually attack if you're not scented as them. So there's like a lot of the biology behind some popular games is correct. Other ones, it's like very fantastical, right? And it's like not actually going to inform you of any of those components. Which,
and I think having, I think you'd probably agree with this, Bullet, but having like aspects of realism in a game like that, if you're portraying something from the natural world, be it ant behavior or dinosaurs, basing that on reality, I think makes for a better game, because it feels just viscerally more absorbing. Like, you see these things and you go, oh yeah, that makes intuitive sense. It feels real, and it makes for a more engaging experience, I think. And I wish certain people would understand that about dinosaurs. Like, the way to make better dinosaurs, and a way to make a better film or video game with dinosaurs, is to make the dinosaurs more authentic, you know? Do you feel the same way about, ants and stuff like that?
Yeah, I mean, there's no reason to change it. Like, well, it's depending on the game that you're going for, right? There's gonna be cool stuff from it. Like, I get if you're going for an ant monster, it's like, then that's a different style of game. But if it's something like the Tycoon Simulator, right, where it's like the whole point is the animal, I think maybe it's just more work to make it realistic and actually put in the research versus just like, I can grab from what's in the Jurassic Park movies, and that's easier to just impose in. And because like the tech here, right? Like we were talking, it's not just for CGI in movies, but it could be used in video games where they do this kind of mapping as well.
Absolutely, yeah.
Heaven forbid you could learn something from the game too, in terms of the accuracy behind it.
I think it kind of goes back to something that I think in like art education, there's this idea that you kind of have to know the rules for how something works so that you can break them in an intelligent way and make good art. So it's like when you have an understanding of something, you know when you can deviate from reality, and you do it in a very intentional way that makes the thing better, rather than just like kind of being lazy about it and just like not caring about the rules, so you break a bunch of them. I don't know, maybe people in chat can relate to that, but like knowing the rules so you know how to break them better, and doing it in a really meaningful, thoughtful way, I think it can lead to really good art, good meaning.
No, absolutely. I think it's the same. It's the same principle in genetics, right?
Yeah.
How can I break the system to figure out how it works? It's the same way of like, how can I, learn how this works, and then I adapt and add on augmentation for the purpose of the game, versus just we're just loosey goosey on everything.
Yeah. I like that. Yeah.
Awesome.
Well, shoot. On that note, I should probably head out.
Danny, thank you so much for your time tonight. I love these crossover streams, and I think it was y'all, we didn't even chat about it. We sent each other our papers. Both of us picked the methods paper today. I'm just like,
Yeah.
our brains are synchronized up.
Definitely on the same wavelength there. Yeah.
I thought that was a lot of fun, like between the new genetics tool and the new tool for measuring like walking behavior. It's, I mean, at this stage, we can make an animal, Danny.
Sounds good. Hey, at TwitchCon 2024, let's bring something on the stage at the science panel if we do that.
Absolutely. Guys, please go follow Professor Danny Anduza Paleontologizing. Right now, if you see a heart at the top of your chat, you're not doing life right because you haven't yet followed him. Hit that follow button. The name's in the episode description. He's our reason for being here on Twitch, and thank you for everything you do, Danny. You are an inspiration to all scientists to do better, and we couldn't be here without you.
Likewise, Bulent. Holy cow, I really appreciate the opportunity to have these chats, and thank you for always being so quick about like finding the papers. It's always before I do every week.
I just know how busy you are too, and I'm just like, let me. I want to get them to you by the weekend, but I'm like, I always end up like running over.
I would not be capable of doing this. So yeah, I don't know. I don't know if this paper was published over the weekend. So like, I'm often just snapping things up like day of that just dropped.
Yeah.
But anyway, I appreciate the opportunity. Thank you for inviting me onto your channel as always.
Always, thank you. What's coming up on the rest of the week to get the party people excited?
As always, we've got Thursday Birds Day tomorrow. We're going to be talking about how birds are living dinosaurs. We're going to be sharing bird pictures from the community posted to the Discord. And then at some point, we might also watch that new documentary with David Attenborough about the sea monster, that big short-necked plesiosaur. You know, a big flesh-eating aquatic animal bigger than a T-Rex from the Isle of Wight. So, yeah, that's gonna be a ton of fun.
That'll be
We might do that tomorrow. We might do that Friday. We'll see.
All right, guys, tune in. Plesiosaur was my favorite dinosaur until I found out it's not a dinosaur. Now it's just my favorite aquatic creature during that era. And my picture books were wrong, but it's fine. We don't know what's correct. Danny, thank you again, my friend. Have a lovely dinner, and I'll see you tomorrow. Guys, Monday, Tuesday, Wednesday, Thursday, Friday, around 5 p.m. Eastern, Danny goes live, and usually for like 4, 5, 6 hours sometimes teaching us about science. So please go follow him, and I'm always in the shadows there. I have him tabbed up hours before he goes live because it's my highlight of my day. Thank you again, Danny.
Appreciate you, Bud. I'll see you later. Have a great rest of your stream, and take care, everybody.
See you tomorrow, Danny. All right, y'all. That was the lovely Professor Danny Anduza.